full screen mode?
Yeah.
Okay.
Can you, are the slides moving? Can you check if you...
Yeah, just a moment. I'll put it on screen share mode, and this is my second slide. Are you able to?
Okay.
Yeah.
Okay. Great.
Okay, so I think we can start. So good afternoon, everyone. We have a lot of people from the GNE myopathy community here, and scientists and doctors here with us today. And it gives me great pleasure to welcome you to the second day of the annual conference of World Without GNE Myopathy. This is our sixth annual conference. And I'm so grateful to all the speakers and participants who are here today on a Sunday afternoon to talk about rare disease research and development and GNE myopathy. And it gives me great pleasure to invite our first speaker for today, Dr. Monika Pahuja, who is a scientist at the Indian Council of Medical Research and is doing a lot of very good work with respect to rare diseases. And she's taken time out to address us, and I'm really excited. I think we're all really excited to hear from you, Dr. Pahuja. So I will hand it over to you.
Thank you, Dr. Shilpi. Thank you, sir. Thank you for inviting me. This is indeed a pleasure to share what ICMR is doing, and more of a, I would say I'll take it as a platform to give a message to all the researchers who are working in the area to take advantage of what is right now available so that we can move faster, because as we all know that we started a bit late on working on the rare diseases, but nonetheless we are now working in a mission mode way to look at the development of newer therapies and to give a holistic care for the patients suffering with the rare diseases. So I just would like to share my screen for my slides. Just give me a sec. I am doing that from my side. So I hope my screen is visible. My slides are visible to all.
Yes.
It's in the screen share mode. Okay. So I'll try to give as much information as possible when it comes to at least ICMR's not only initiatives but the way out, the help, the platform, the way we are trying to engage researchers, and we are trying to bring collaborations so that more and more can be given back to the society, and specifically the patients suffering with the rare diseases. We would like to come at the fastest rate possible for the drugs, for the therapies, devices, anything and everything possible for the rare disease patients. With this, I would like to first give a brief outline that there's a national consortium for research and development on therapeutics for rare diseases, which is as envisaged in the national policy for rare diseases. It was mentioned there in 2021, and in 2021, I think October or November, this consortium was formed by the ministry and all the major stakeholders, including ministry itself, DBT, DST, ICMR, CSIR, patient organizations, DGHS, DCGI even. They're all part of this consortium. So we are trying to make more, and although consortium is not earmarked with any fund right now, but we are trying to make as much as funding can be given through ICMR for rare diseases. Good thing is that rare diseases is now identified as the priority area while looking into the priority area of ICMR, because nowadays ICMR gives out calls for specific areas. So rare disease is one of them. uh, in ICMR, while brainstorming on the rare diseases, we looked on the matter that because 450 around 450 rare diseases are identified in the country, so we cannot work on all the rare diseases at once. So we try to identify the rare diseases, few of them for which number of patients is highest, or for which some therapeutics are available worldwide, which can be brought in our country. And so that's how we prioritize DMD, SMA, Gaucher, Pompe, Febrile, Niemann-Pick, alkaptonuria, and familial hypercholesterolemia, for which we try to give more and more calls on. But it's not that our purview is limited to these conditions only. We are ready to take anything for which any proof of concept available to the next level as soon as it comes to us. So, talking about the ICMR initiatives, ICMR in 2019 started work on rare diseases with a task force on rare diseases, wherein it was an open call sort of, because it was not concentrating on therapeutics only. So, diagnostics, biomarkers, even the genotype variants are the kind of projects we got in this. So, because it was type of scattered, and we didn't get much in terms of the output, so that's why we thought of bringing out a special call for rare diseases, which was specifically looking into the therapeutics. Herein, this call came out in 2022, and now all the projects are ongoing in this. Then after that, we thought of bringing the industry in. So we gave an expression of interest wherein we try to pitch in the industry, but within our ICMR system, there is no provision through which we can fund industry or any private organization. So here in this expression of interest, we try to make it like this, that this industry needs to have any academic collaboration, or even if industry is taking bringing any product by themselves, then ICMR will fund only the clinical sites. ICMR will not fund the industry, but will directly fund the clinical sites if a clinical collaborator is, if a clinical study is involved in this, or it can fund. Okay, or we can fund to the labs which are working on the animal experiments and all. So, in continuation to this, because ICMR identified that there are cell and gene therapies which are coming in a great way, especially for the rare disease patients. So, we try to look more into gene therapy as well as the regulatory pathway, because this is where most of the academic researchers, either they are not very well versed with the regulatory pathway, or they are not having infrastructure or support with them to go through this pathway. So we are holding regular seminars or meetings with the CDSU officials, wherein they come and they give specific answers to the query of researchers for their own products. uh, Coming to the main role of ICMR, which is funding. So currently, I would say I'll take an opportunity to urge all the researchers to um, uh, to motivate them to please submit proposals. Small grant, which is now up to 2 crores, is open. This call is open, and rare diseases is one of the priority in this. And this call is open till April 1st. So I would say researchers to please submit their projects. And this small grant is basically looked at the to generate you know to generate data in a pilot study mode, or an initial discovery project kind of thing, which comes under small grant, and this covers up till 2 crores. Next is the intermediate grant, which is from 2 to 8 crores, which leads to the basically the development of the full of any product or therapy. And this call is also open, and the last date is 8th of April this year. And the last is the which is mainly a collaborative research. It's called Center of Advanced Research. This gives a broader opportunity because this may include multicentric studies. And it gives a fund of up till 15 crores, and this is mainly for 5 years. And this is the website through which these all can be looked into. Coming back to the initiatives, one area wherein ICMR is giving a lot of thirst is drug repurposing for rare diseases, because of the very obvious reasons that it brings faster availability. It does not include mainly the patent problems, because the biosimilars or the drug is already in the market since long, so it reduces the time and cost required for a new drug development. And it also improves the quality of life for patients, for which a faster delivery is very much required. It also leads to the affordable treatment options, because these drugs are available at a very cheaper Most of these are available at a cheaper rate, and we try to inculcate them in the system with a second indication for these drugs. So a lot of work is going within ICMR for this initiative, and we are trying to take more and more drugs for the clinical trials for the rare diseases. And although we tried with the priority 7 rare diseases, 7 or 8, I just told you. But the thing is, we are taking as much as possible. We are not only restricting ourselves to those priority disorders. The next move which ICMR took is collaborating with CDSCO, because CDSCO and ICMR now have a kind of collaboration wherein ICMR scientists are going to CDSCO and giving them inputs on specific matters which are required. This could be small molecules, biosimilars, or cell and gene therapy, or medical devices, because all these four types of things are very much required in rare diseases. So a regulatory path is also trying to be, clutter free for the academicians, and they can anytime come to ICMR and ask for that help. And I'll just give in my next slides that how that can be done. So this is the another initiative of ICMR called MedTech Mitra, wherein we have identified this Death Valley where the researchers, the academicians, does not get an idea how to move forward, or they does not have an infrastructure to move forward. So from proof of concept to coming to the market, all the steps are covered in this MedTech Mitra. We are providing regulatory support to the researchers. We are providing them for preparing dossiers for clinical studies. We are providing them validation studies for their any type of devices. We are providing them through our DHR initiative on health technology assessment of their, if any technology is available and they want to bring it to in the policy. So that is also available through this. Another this is all how it looks like if you go to the website of ICMR and click on the medtech mitra link. This is how you can reach us. It's a form, a very simple form. You can fill that form and that directly comes to us, or you can ask for anything related to the drug development, which may include regulatory strategy, which may include facilitators, policies, health technology assessment. Clinical evaluation is done through our intent network, which has 100 plus sites now, and we are trying to include all the center of excellences of ministry on rare diseases within this network. Next, and the most recent, is the Patent Mitra, which comes in the public yesterday only through our Honorable Minister of Health and Family Welfare. It comes with a vision that nowadays researchers gets bogged up and isn't able to take their products, their molecules patented. So ICMR will be providing all the assistance to any researchers. It has two or three wings. One is which come the patents or the products which come out of the which come out of the projects which are funded by ICMR, and another is anyone who is having any product and facing issues with the patent can come to ICMR, and we will be happy to take up this for their patent protection. So these are the center of excellences, and we are trying to work with all these centers for our clinical trials, and we'll try to, these are the will be our first choice for taking up the clinical trials. But we are not restricting ourselves to this, because if you know that number of patients are really less for clinical trials in the country, so we will be taking uh, private centers or centers beyond this center of excellences for our as our collaborator for the clinical trial. Thank you. And if anyone has any issues, any products ready, want to have some support from ICMI in any form, in terms of funds, in terms of patent, in terms of how to take it forward, preparing dossiers or anything, they can write to me on this email ID. Thank you. That is all from my side, sir.
Thank you so much, Dr. Pahuja. It's really, I mean, this is really a new, I think, a new space for India where, this level of work is being done for drug development and rare diseases, and the kind of support that ICMR is providing. It's really nice to see that. And once again, thank you so much for, explaining all the work that you're doing so clearly and so well. Did anyone have questions? If you do, please raise your hand.
So I believe there's something come up in chat box.
And that I think we will ask from the doctors.
Yeah, I believe that is not related to this particular talk.
Yeah.
I'm happy to take up any questions, even if after this seminar or after this, I have provided the email ID where anyone can reach to me and can write to us for any help required, which is within the purview or within our scope. We'll try to handle as early as possible.
Um, Dr. Monica, maybe I can. Good afternoon.
Good
Uh,
afternoon, ma'am.
about the centers of excellence, I'm speaking on behalf of the patient groups. So there's a lot of patients are saying, that it's very difficult to navigate through the centers of excellence. I mean, it's because the doctors really don't have so much time, and the rare disease patients, they are kind of lost. They don't know exactly how to approach and so on. So maybe you could just you know, uh, maybe a meeting with the patient groups, not GNE myopathy itself, but a lot of other patient groups. I'm talking on behalf of them. Uh, some kind of a information, crosstalk between patients and the COE. I think that is needed to streamline the use of the COE itself.
Sure, ma'am, I do agree. And if I don't know whether the group is aware or not, but the number, email IDs of all nodal officers of these Center of Excellences are available on the ministry's website. If required, I can provide also. Beyond that, once you get in touch with the nodal officer, they will be able to guide the patient further how to take it.
Okay, so the nodal officer information is there on the website.
Yes, ma'am. Their mobile numbers, their email IDs are available.
Okay, okay, okay. So that would be a good place to start first.
Sure.
Okay, thank you.
Thank you.
Dr. Pahuja, Dr. Vishnu is asking, do you think rare disease focus has neglected adults compared to pediatric populations?
Uh, I'll say today the focus is pediatric population. Yes, we have not taken yet, we have not reached a state yet where we are talking more about the adult rare disease cases. So, although we are not, we do agree that we have not reached there, but yes, we are thinking in this direction also. We are thinking that now it's the high time to include adult population and their problems in this mission.
Thank you. Any other questions?
Just Monica, there's a simple. Does ICMR has a target or something saying that we want to come out with so many drugs in next 5 years or some such thing? Are you working
Yes, sir.
that kind of
Yes,
a
sir.
target?
We have taken up this challenge of giving out 10 drugs in next 5 years. And
Oh,
that 10 drugs is not like newer therapies. They can be repurposed drugs.
all
So we have
right.
taken
We
this
hope that
challenge.
we can be one. At least
Yes, sir,
think
sure.
of-
That's why we are taking everything which is coming to us, giving us a very serious look. And if it shows some chances of going forward, any proof of concept is there, we are trying to take it.
Yeah.
Just wanted to thank you for the Mitra schemes. I think it's an excellent initiative,
Thank you,
and
ma'am.
it
Thank you.
will
We would,
it will really help us, who are groping in the dark sort of a situation. So now we know where to go. Thank you
yes,
for that.
ma'am. Definitely. Thank you.
And I just a very random question, but other than this, is there any thought from the government or ICMR on, say, compulsory licensing for some of these rare disease drugs or other ways of like making them
Mm.
available?
It's not compulsory licensing among one of the initiatives which mainly comes from the high court order. The high court order through, you must be aware, there were many court cases of DMD and they were asking for drugs. So court made a committee called NRDC. So this committee has done a lot of effort in this direction for making drugs affordable for the country. So through this committee, wherein ministry is mainly taking an initiative, they have reached out to almost all the drug manufacturers in this area and asked them to either make it affordable through uh, the ministry is ready, government is ready to uh, give them any facilities if they want to manufacture it here, or they want through their normal route of having some sister company which does namely marketing here. So it is also ongoing activity, and it is done side by side. Apart from looking into newer drugs, the older which are available anywhere in the world, government is trying to bring them in through affordable pricing, either through manufacturing or through negotiations. All the routes are kept open, and government is working on that.
Because we also have a number of now and the next few some of the speakers in the following sessions, they are involved with developing some chemicals that can help that can help us, um, but we have to, I think, just figure out how that can be then
Yeah,
manufactured.
and just one thing I would like to give a message to the researchers, that if they have any in-house research going on, wherein they have any proof of concept, to come to us to make it. We have this mission to bring it at the earliest possible to the patients. So for that, we try to bring all the components wherein normally researchers fail. We bring all those experts at one place, and we are even nowadays providing, if you say you have a molecule but you don't know where to, I have tested it in the animals and efficacy is there, but I don't know how to go forward. So I'm myself is giving collaborators, and you must be aware that ICMR has now come up with the institute called NARF, that is in Hyderabad, and that is mainly for the safety toxicology of all the products, vaccines, devices. And we ICMR has already initiated the process to get it GLP accredited through DST and GCMA. So we are trying to bring it risk free for the companies as well as for the researchers, so that we are trying to pitch in at every stage wherein they find it difficult, because safety pharmacology studies or GLP toxicity studies are the most heaviest on the pocket for any researchers or even for the companies. So we are de-risking them. We are giving our center to do all these studies. Our center is equipped from rat, mice to even monkeys. They have all the facilities available for vaccines, for drugs. So everything is put in place. It's just that we need more and more good researchers to come and collaborate and come and bring their products, so that we will be able to direction them or bring them to the market.
That's very, very heartening to know. Thank you so much, Dr.
Thank
Pahuja.
you.
That's like very, very positive from your side. And it gives us a lot of hope. And I think we will be working with you closely. So
Thank you.
look forward to more
Sure.
collaboration.
Looking forward to it.
Thank you, Dr. Pahuja. Thank you so much.
Thank you, Shilpi. Thank you, sir.
So now I would like to invite our next speaker, Dr. Satish Khadilkar, who most of us in the GNE myopathy community know very well. He's a very senior neurologist who has seen and diagnosed a number of people with GNE myopathy. He is a consultant neurologist at Bombay Hospital. So I would like to invite Dr. Khadilkar to give the next talk. I believe his talk will be recorded, but he will be joining us for question and answers.
Good evening, everyone. Firstly, let me thank Dr. Bhattacharya and Dr. Sudha Bhattacharya for asking me to be here and to hold this lovely meeting year after year for the GNE myopathy patients.
We can't hear, we are not able to hear. There's some issue with the audio, I feel. Is it just me?
No, we are not able to hear.
We're not able to hear, Baba.
Nothing is coming.
Good strength in the quad.
Yeah, the audio is not coming properly.
So how does this
Audio is not here.
present? Clinic?
I think there's some issue with the internet on your side.
My side. You're sharing.
Maybe play it again. Otherwise, I will try to play it.
Yeah, why don't you try to play?
I don't have it, so
I'm hearing the sound, but I don't know whether it will get to everybody or not.
Good evening, everyone.
Can you hear it?
Firstly, let me thank
Yes, sir, it's audible, although a bit low, but yes, audible.
for asking me to be here and to hold this lovely meeting year after year for the GNE myopathy patients. Today's topic of discussion is management of GNE myopathy. But let's see a little bit about GNE myopathy before we proceed. So as you know, and as other speakers have been telling you, GNE is an ultra-rare disease. Whenever you're talking of a rare disease or an ultra-rare disease, it goes without saying that very few people will know about it. Medical practitioners also at times may not know about it. General public doesn't know about it. And therefore, the sufferers find it very difficult to reach a diagnosis, then find doctors and patients who have suffered from the same illness, and then try and find a treatment. So these are some of the problems of ultra-rare diseases. Moreover, this is seen in only some parts of the world. So when a patient relocates to another part of the world where GNE myopathy is not common, he is likely to face more difficulty. This condition was originally investigated in Japan and Israel. And now we know that there are patients in other countries. We also have a lot of patients of GNE myopathy in India. And the work on GNE myopathy in India has been done by the groups in Bengaluru, group in Mumbai, and there is an occasional paper from other places in the country, such as Chandigarh, where they called it HIBM. So these are the two original investigators for this disease. On the left of the slide is Zohar Argao. Who investigated patients in Israel, and he focused on the good strength in the quadriceps muscle, whereas other muscles were getting weak, and therefore he called it a quadriceps sparing myopathy. Around the same time, Ikuya Nonaka, who is on the right of the frame, was investigating patients who had a distal weakness of the tibialis anterior muscle, resulting in a foot drop. And that was then called Nonaka myopathy. So these were the two original names, quadriceps sparing myopathy and a distal myopathy. And these two are the main investigators who studied the GNE myopathy. So how does this disease present? Clinically, patients have a foot drop and their feet start dangling. Since the foot cannot be cleared off the ground, they tend to hit objects, they tend to get imbalance, they fall. So change in the gait and easy falls is one of the early features that you see in GNE myopathy. Along with that, the proximal muscles also become weak, but the quadriceps remains strong for a long time, as was detected by Argao, the quadriceps sparing myopathy. As the disease goes on further, it also goes to the trunk, and the trunk becomes weak, the core muscles become weak, and later on, in some patients, the hands and the fingers also become weak, and sometimes the first dorsal interosseous gets wasted. Now, when you're investigating such a patient, you will see the serum CPK to be elevated, but not very high. Like some myopathies like dysferlinopathy or calpainopathy will have a CPK of 5,000, 10,000 or like that. In this condition, the CPK is usually under 1,000 international units, so 700, 800, like that. And the electrophysiology, which is one of the popular tests for diagnosing myopathies, shows myopathic electrophysiology. And biopsy shows rimmed vacuoles. Now, this is one of my first few patients' MRI. MRI is a newer test that you can use to find out which muscles are affected. And it's a non-invasive test. So some people like to do an MRI. And you can see here, on the left panel is a thigh section. On the right panel is a leg section around the shin. So on the right panel, you see that the bright spot here where my cursor is the bone. And on the medial side of the bone, there is tibialis anterior muscle. And you can see a change in the signal in the tibialis anterior muscle. So this patient, you can infer, must be having a foot drop. In addition to that, when you go to the thigh section where my cursor now is, you see very good quadriceps muscle, and you see a lot of involvement of the hamstring muscle. So MRI can be useful in making the diagnosis by showing signal changes in the tibialis anterior muscle and the posterior part of the thigh and sparing of the quadriceps muscle. So MRI patterns now have been well described in this condition. When you do a biopsy, in the biopsy you will see rimmed vacuoles, which is the major finding in GNE myopathy. And every high power field will show you some vacuoles. Where my cursor now is, you are seeing a vacuole. One more is here. One more is here. So these vacuoles, which are rimmed, that's a pathological hallmark of this disease. Mind you, vacuoles are also seen in other diseases, not only in the GNE myopathy, but in patients with GNE myopathy, you see them. Now, this is the era of genetics, and in the last few years, information on genetics of so many different myopathies is available, and GNE myopathy is not an exception. Here is a very beautiful chart from one paper from Tahsin Muzaffar from San Francisco, where he has shown, when he's collated data on the GNE myopathy genetics. And as you can see, some genetic faults are more common in some parts of the world as compared to others. So here you see that in the Japanese individual, a particular mutation is common. Chinese, another one is common. Bulgarian Roma and Rajasthan region of India, they have another mutation. India has VAL 727, and so on and so forth with the Middle Eastern and the Northern British population. So depending upon which part of the world you come from, the common genetic mutation changes. I would like you all to see this paper by Dr. Bhattacharya, authored by Sudha Bhattacharya, the first author, where they collated all the known genes that were known in GNE patients through a patient questionnaire, and they found that VAL 727 is one of the most common genetic alterations in India. In the western part of India, when we were studying the GNE myopathy, there were some patients who came from Rajasthan origin. And these people harbored a particular mutation which they shared with the Roma or the Gypsy people. And this is also of interest because it is not, it's believed that the Roma originated in northwest of India and then went out to various places in the world. And the origin of the Eastern European and European Romas is from Rajasthan. So this study sort of gave another reinforcement to the idea that Roma have originated from Rajasthan and that they have a particular founder mutation, which is usually homozygous in these patients. So with this information on the GNE myopathy, let's come down to the management. Management of any chronic neurological ailment is very difficult because people are suffering for a long time and their reactions have also changed. So the practitioner has to be very, very careful. Now, let's take one by one. First is diet. In diet, balanced diet is very important, which contains proteins, carbohydrates, and fats in a good measure, along with the vitamins and minerals. It is very, very important for the patients of myopathies, any type of myopathies, to realize that they should avoid weight gain. Weight gain is an enemy because muscles have to carry that much more weight. In our traditional way, when somebody is weak, into inverted commas, weak, the family and sometimes the elders and well-wishers will say, food has to be increased and give this and give that, and give nutritious food. So patients are put on excessive nutritious food and they go on gaining weight. And once they gain weight, their mobility actually starts reducing. So avoiding weight gain is a very important factor for patients of GNE myopathy and all myopathy. The other thing that I am mentioning is that these days proteins are the flavor of the season. We sort of want everyone to have good proteins, and we're discussing proteins, and we have charts as to how much proteins are required. I find in clinical practice that patients are having an overload of protein, and that should be avoided. One should take proteins in adequate measure, a little more than what one would normally take because there is a muscular dystrophy. But please remember that proteins have to be converted by the kidneys as they go out, and the kidneys don't like too much protein, and you can start suffering if you take too much protein. So please don't take these protein supplements that are available in the market for health, in the health industry, the bodybuilding and those sort of things. Please, if you take it only with the advice from your doctor. After diet comes exercise. What's the role of exercise? As we saw in the clinical features of the GNE myopathy, some muscles are weaker than the other, and because there is an imbalance, the joint doesn't move through a good range. So one aim of the exercise is to maintain the range of motion across the joints, where all the joints should be moved through their full range to try and maintain them. The other part is the strengthening of the muscle, where you do resistance training so that the muscles that are good become stronger. Now again, please remember that do not overdo. When we make a diagnosis and we start telling people, please, you must exercise, they overdo it. They start pushing themselves beyond 100%, and that is definitely damaging to the muscle. So a general guideline that I give is 80% of your strength you should use, not more than 80%. So whatever you're able to do, you do 80%. If you can do 10 repetitions, you do 8. If you can walk for 10 minutes, walk for 8 minutes. So this I call the 80% rule. Don't go beyond the 80%. That can be damaging. Then the prescription. Specific modifications we will come to in the coming slides. But in Indian people, particularly those who are vegetarian, vitamin D and vitamin B12 deficiencies are very common. And therefore, always check and make sure that your vitamin D and B12 is not low. If it is low, take replacement and keep them at a good level. India is a country where different systems of medicines are prevalent. You got allopathy, you got Ayurveda, you got Unani, you got homeopathy, Siddha medicine, and so many others. So often when we tell that in allopathy we do not have very good treatments, people go to other forms of therapy, alternative forms of therapy. We do not know too much about them, but we know that some of the methods like having metallic oxide in Ayurveda can be negative, can have a negative impact. So please, whichever alternative medicines you are taking, make sure that there is a senior and an experienced doctor to whom you have gone. And if you wanted to try, then you try along with him. So prescriptions actually are very short or brief in this condition. In rehabilitation process, it is very, very important to make things user-friendly. So designing the doors for wheelchairs to pass, automated door knobs so that you don't have to squeeze it open. Various remote control devices which sort of work the curtains, the television, fans, the lights, things like that. Adjustable beds, the height of the bed, the inclination of the bed can all be monitored now. Then slings kept outside the bath so that you can use them when you're getting out of the bath, out of the bed, and railings in stairways. These are some of the common home accessibility aids that one can use to improve the functioning and to avoid falls. Avoid falls is another important thing, because if you fall and have a fracture, there can be trouble. To increase mobility, you can use a crane, you can use crutches, you can use walkers, you can use braces, you can use wheelchairs. And this along with your physiotherapist and the orthotics doctor, you will need to navigate as to what is most useful for you. One has to pay attention to mental health. As I told before, these ailments are chronic. People are not able to do everything that they want to do, and therefore anxiety and depression creeps in. Strengthened social support networks assume a lot of importance. And patient groups and organizations, as we know in this day and age, are very important support systems where one person can tell how he overcame the difficulty that he was facing, what aids did he use, to whom did he go, and so on and so forth. So I think tight patient groups and organizations are a source of lot of information to patients in such rare diseases. They also strengthen the mental and the physical strength for these patients. If the anxiety and depression is too much, then one could resort to taking some form of treatment, pharmacological. Now let's come to specific therapies. As you know, the brunt of the research has been on sialic acid. And you can see in this diagram, glucose is converted into UDP, G1C, NAC, which is the sialic acid. Then that gets converted by two rate-limiting enzymes into MANAC, and then MANAC6-phosphate, and then neuroacetamino acid, and then to sialic acid. So glucose is converted into sialic acid by two major enzymes, as this diagram shows. Then it enters the Golgi apparatus, and then it comes out to the Golgi as sialylated glycoprotein. So that's the cycle in normal individuals. Now we know that GNE myopathy is related to sialic acid, but the relationship between sialic acid reduction and GNE myopathy weakness is not very clear. There are people who don't have reduction in sialic acid levels but have a GNE myopathy, and vice versa. So it's not a linear one-to-one relationship. Also, the GNE protein quantity may not be reduced in some GNE patients, which is another major variation. And as you know that some patients have thrombocytopenia or platelet dysfunction, where sialic acid is important. And we also know that amyloid beta peptide accumulates in vacuoles. So even though the thrust has been on sialic acid, there are other things to consider, and sialic acid and GNE myopathy is not a one-to-one relationship. So what are the treatment trials available? Intravenous immunoglobulins have been tried. MANAC has been tried. Then per-acetylated MANAC has been tried. But these two trials are on mouse model. There are some human trials, and recently the medication acinoble, which is acetylnuraminic acid, has been available in Japan in the last 2 months. But at the moment, I am told that it is going to be available only for people of Japanese origin because they have only a limited supply. And there is a Korean medicine called 6SL is available. They are both external supplements of sialic acid, and it remains to be seen how they are actually helping patients in the long run. But we have tablets available, so now in a year or two, we will have some data on how these tablets are helping people. Are they reducing the rate of progress? Are they stopping progression? Are they reversing anything? And then clearly more studies are required before we can settle down on a good therapy of sialic acid. So at the moment, this condition needs management. It cannot be reversed, but in time to come, we may see that as well. Thank you very much for your attention.
Thank you very much to Dr. Khadilkar for that exceptionally informative talk. Is Dr. Khadilkar here to answer questions? I believe he's not here. So I think we can then move on to the next talk.
I'm ready.
Thank you so much. I'm just going to. I don't see Dr. Khadilkar is here. I think he has not been able to join. So the questions that we have for him, we can take up in, I think maybe Dr. Vishnu could answer them later on. So I think then we could move on to the next talk, which I think many people are waiting for. So I would like to welcome Dr. Jin-Hong Shin who's an associate professor at Pusan National University, Yangsan Hospital, Korea. And he's joining us on this Sunday evening, I guess, your time from Korea. Thank you so much for joining us on Sunday and for taking time out on a holiday to address our patient group. We really appreciate it. I would like to hand over the platform to you. Thank you so much, Dr. Shin. Are you able to share your slides?
I'll try to share my slides. Wait a moment. Can you see my slides?
Yes.
Okay, let's go ahead. So first of all, it's my great honor and pleasure to be invited here. Thanks for having me. We have been working on the 6-silent lactose supplementation in GNE myopathy for the past few years. And our recent data has been published in 2 separate publications. And if anyone who needs more further information, you can just take a look at our publication. So I wouldn't have to explain anything about the GNE myopathy because Dr. Khadilkar has made a great introduction to the GNE myopathy. But one thing we have to remember is that the GNE myopathy is actually the problem with the GNE gene, which is the key player in the sialic acid biosynthesis. So the pioneering research by Dr. Malikdan and Dr. Noguchi and Dr. Nishino has proven that either sialic acid itself, MANA, or sialic lactose can help the progression of the disease in the GNE myopathy mouse model. And among the 3 of them, the sialic lactose was the best one, which can ameliorate the myopathic phenotype, even in the symptomatic GNE myopathy mice. So we jumped to the sialic lactose. We use the 6-sialic lactose. There are 3 sialic lactose and 6 sialic lactose, but we choose 6 sialic lactose. Sialic lactose is actually, as the name implies, it's the sialic acid plus lactose. It's different from MANA or sialic acid itself. It is absorbed as an sialic lactose is most natural compound to supply sialic acid to the babies. So it is abundant in the milk. So we did on clinical trial. It has been some years ago. We had a short period of placebo-controlled, 12 weeks of placebo-controlled period, and the following parts are all the high dose and low dose of sialic lactose, and we measure the power and the imaging. There were some adverse events, but almost everything, actually everything was negligible, self-limited. So we didn't have serious adverse events. And we could see that if we ingest sialic lactose, the sialic lactose level goes up and goes down. And over time, the free sialic acid also goes up. And over the time, over the 12 weeks, there was a great difference between the placebo and the ingested group, that the free and bound form of sialic acid actually goes up. And most importantly, the limb power, the motor power, actually improved after 96 weeks of clinical trial. it is almost 2 years, and you can see even at the end of 48 weeks, which is the end of the first year, we couldn't see great differences between the high dose group and low dose group. But after 1 year, there was difference in the treatment group. So as all of you know, it is a not so fast progressing disease. It is very slowly progressive, but steadily progressive disease. So it is uh, not a good. We need a longer period of observation to see the difference between the treatment and the placebo group, and this really makes a great trouble in the upcoming approval grade clinical trial. Again, with the lower limb power, there was a great difference in the low dose group and the high dose group, but also we could see improvement in all those treatment groups. So it is very rare occasion that we could see any kind of any degree of improvement in the field of neuromuscular disorders, because most of these neuromuscular disorders are steadily progressive, going worse over time. But with our treatment, we could see at least at some part of the limb muscles, we could see the improvement. And this is the MR image. There is no great difference by visual examination, but when we measure the fat fraction quantitatively, there was a difference between the high dose group and low dose group. This is about the fat fraction, so higher is worse. So the low dose group goes higher and the high dose group goes lower. So we could see the difference in the high dose and the low dose group, but we couldn't prove our conjecture because we were without the placebo group to the end. So as a subsequent trial, we devised a placebo-controlled trial, but only for the 48 weeks. And the measurements are all the same. But as expected, we couldn't see a great difference in the treatment group and placebo group. Only on the hand grip, there was a statistically significant difference in the placebo and 6SL group. But this time in the muscle fat fraction, we could see a greater differences in the posterior thigh group. So we could see changes even in the by visual examination. There was a difference between the treated group and the placebo group. And this time we included some kind of more fancier biochemistry assessment. And the left one, the panel A, is the SNA, which actually measures the sialic acid itself at the terminal part of the glycobiome. And the middle one and the right one actually measures the galactose, which can substitute or exposed when sialic acid is there are lack of sialic acid. So for the panel A, higher is better. For the panel B and C, lower is better. And we could see exactly what we expected for. So our 6-SL supplementation, we could see that it changes in the content of sialic acid on the cell surface, and it has an impact on the MR imaging, and also it improves the muscle function. So all of you may be anxious just to how to get the 6-sialic lactose, but I'll tell you some stories, but you have to be a bit of careful. So the availability of 6-lactose, as far as I know, 3 companies are running to supply lactose to the general public. The 2 companies on the left side, the California and the other company, are on sale in internet, but I'm not working with either of them. So I don't know them, they don't know me. So I cannot guarantee anything about the quality, purity, or the efficacy. So it'll be a good chance that if the patient support group like yours can buy some of these compounds and test it in the certified labs, that how pure it is, a 6-lactose, or what kind of impurities they have. So and it'll be better to share among the communities that if it is good enough to be ingested as a food supplement or as a drug. But at this moment, I cannot say anything for or against these 2 compounds because I'm not working with them. The one on the right side, Neurology, is the one I'm working with for the past few years, and currently they are not producing for the patients. They are in preparation of the approval grade clinical trial. So they are making the 6-lactose into the rare disease drug, which is really important. But for that, they need many kinds of steps, like the previous presentation, we need a toxicology and the approval grade clinical trial to show the real efficacy of the 6-lactose. The data I have presented for the past few minutes is on pilot trial, so it is not so a good quality like an approval grade clinical trial. So Neurology is really working hard to prepare the approval grade clinical trial, but it takes time. So in the meanwhile, I know the patients are so anxious and so deliberate to get any kind of drugs for their disease. So, but I cannot say to buy this or not to buy this. It's on your discretion. So anyhow, it is my obligation to tell you that there are some kind of options, at least you can choose. And also, I'm working hard with Neurology to make this 6-lactose to be the real approved drug for all GNE myopathy patients in the world. So I have to acknowledge that this study has been couldn't be done without the help of all these people in the hospital and the people in the company, and also many patients in the Korean GNE myopathy patient support group. And you have to you will know that it is how it is like to be assigned as a placebo group for as I have pointed out in my previous slides, that it is a steadily progressing disease, so it doesn't make a great difference over 1 year. So to make the difference between the treated group and the placebo group, we need a long time of observation. So at some point, the Neurology and myself will maybe we'll reach out to the Indian people to incorporate, to be enrolled in the clinical trial. And at that time, we will also need your help to make this drug into the reality. Okay, this is my last slide, and I'm ready to get any kind of questions from you.
Thank you so much, Dr. Shin, and we would be extremely happy to collaborate with you and Neurogene with respect to any future trials that you would like to do. Please feel free to reach out to us. We would be happy to participate in such endeavors. There's a few questions and one comment. One patient is saying that they are benefited from taking 6SL. Neetu's asking, how can we check sialic acid levels in the blood or GNE gene protein in the blood?
We can measure it in the laboratory, but for the it is highly recommended to measure it on a certified laboratory. You may be able to find some laboratory who can commercially and independently measure sialic acid level or the 6-silyl lactose level for the patient advocacy group. So it'll be better to organize a small task force to take the drug and get the blood measurement and have it measured in the independent certified laboratory. You can reach out for the national laboratory, like the first presenter.
Dr. Devinder Kaur is asking, what is the age group of the participants in your study? Is it beneficial in case patients already lost most of the muscle mass?
No, for the first one, we included the only walking group, and for the second one, second study, we included even better moving patients, because there isn't steps in the clinical trial to prove the efficacy of the drugs, but the higher chance is for the patient who has more muscle. It is just a speculation. We don't know if it's gonna work on the more advanced patients or not. But for this time, we focused on the walking patients.
Thank you so much for that. Dr. Sudha Bhattacharya is asking whether California supplements. Number of questions are coming on the chat. Okay, so Michaela's asking whether, Dr. Shin, thank you for noting the importance of doing extended clinical trials. I think working on the design of the trial will be very important at this stage. Will you consider using the Bayesian model applied by NIH to have a powerful statistical?
Definitely. That's something we have to adopt. For the we are at this stage we are doing the toxicology study, but in a few months we will try to contact FDA, US FDA, and at that moment we will have to decide what kind of statistical measurement with statistical method we have to use for the real approval grade clinical trial.
So Dr. Sudha Bhattacharya is asking, she's saying that the California Supplements company is quoting your study to market their product. Are they getting any backing from the patient group in California?
I am not aware of anything about the California company.
So one patient is saying that they got hemorrhoids in both the leg and both their legs were swollen after using 6SL. and once they stopped it, they returned to normal. The swelling of legs and the gastric side effects, is that something that you've observed?
Well, I don't think hemorrhoid can be any kind of a side effect that related to the silylactose. But well, for the California or the other company, the 6-silylactose, I cannot guarantee the purity or quality of the drug. So any kind of side effect is possible. So that's why I'm urging you to get the measurement of their products in an independent laboratory. So if the 6-silylactose the Neurogene is making is more than 99.9% pure of 6-silylactose. It is already pharmaceutical quality. So as for our experience, we have not experienced any kind of lasting side effects. So only for a few weeks, there was some mild gastric disturbance, like constipation or diarrhea, but all gone naturally, all gone without any treatment.
So there are a few questions on trial design, and even I wanted to ask you, because just from the perspective of people who are looking to design trials for GNE, so Dr. Vishnu is asking, what's the ideal duration of trial to get an unequivocal result? Would you say it's 3 years? Longer duration trials to keep patients in a placebo group would be difficult.
Right. That's the problem. That's the problem we are working on. For our first study, the study period was 2 years. It is just after first year that started to make a difference between the high dose group and low dose group. So our second study, which is 1 year, didn't make a great difference between the placebo and the treated group. So we may need 2 years, and that's really heartbreaking to give the patients the placebo for 2 years. So we are working to solve this problem. According to our first trial, it made an improvement, even improvement from the baseline. So it is one possibility that we will go without placebo group, but I'm not sure the FDA will agree or not. And Neurogene product is still not on the market. They are preparing for the real clinical trial, but they are not selling it to anybody, even to me. They are not selling.
So I just had one last question for you, Dr. Shin. What would you say is the one biggest learning that you've had from doing this trial that you're taking into the next trial that you're planning to do? Like, what should people who are now planning to do trials look out for, things that you think should be kept in mind that are very important?
Well, it takes time. We are sure that this silylactose works to the at least to the motor function, but to prove its efficacy, well, we need time, and we had many kinds of thoughts about our development period, but this is our conclusion that we have to go to the right pathway, the most formal pathway to get the drug to be approved. So Yeah, for the patients, one day is important to any patients, but we will need more time.
Okay, thank you so much, Dr. Shin. There's still some questions coming into the chat, but we are a bit short of time. Would you mind responding to those in the chat itself, and then we can move on to our next speaker? Would you be okay with that?
Well, I'm fine. And also I'm happy to get any kind of questions by email.
Thank you so much. Thank you so much, Dr. Shin, for that very informative talk and for all the work that you're doing in developing 6SL and good luck for the clinical trials and the FDA approval that lies ahead. I think the whole community is behind you and wishing you the very best, and we would be happy to help you in whatever way we can. So thank you so very much. Now I would like to invite our next speaker, who's also an extremely special speaker that we have today, Dr. Wakako Yoshioka, who is a researcher at the Department of Neuromuscular Research at the National Institute of Neuroscience, National Center of Neurology and Psychiatry, Japan. And she's also someone that lives with GNE. And again, thank you so much for being here on a Sunday to speak to us. We really look forward to hearing from you.
Thank you. Thank you for inviting me to this precious meeting. I would like to, can you see my slides? Not yet.
Not yet.
Not yet. Mhm. See my slides and points. Points right now.
Yes, absolutely.
Okay. Okay, I would like to start. My name is Wakako Yoshioka. I'm a medical doctor studying about GNE myopathy, and also I myself is a patient of this disease. So this is a world map showing the founder mutation of GNE myopathy. So as it was introduced in the previous presentation, V727M is the founder mutation in India, and in Japan there are 3 founder mutations, and it is estimated that about 400 patients exist in Japan. And this top figure is of 2019, when Dr. Alok have kindly visited Japan and gave us the lecture, gave the lecture to Japanese patients. So it's an honor for me to talk, giving me the opportunity to talk at this WWGM today. And Dr. Nishino, who is standing next to Dr. Alok, is the person who has guided me to this National Center of Neurology and Psychiatry. And these are my colleagues. And he's one of the researchers who has contributed to understanding the pathophysiology of GNE myopathy. And I would like to start my talk from recent findings about management of this disease. So about progression. In Japan, we have registry of this disease, so registry of muscular dystrophy, named REMD, and it is estimated that about 400 Japanese patients exist in Japan, and 60% of these patients are registered in this registry. And the strength of this registry is that diagnosis is confirmed by genetic curator, and registration form is filled by attending doctor. So the data quality is very high. And every year, patients send us the latest data through their physicians, and it made this long-term observational research possible. So I would like to introduce what we have learned using this data accumulation, since the registry was first established in 2012. So this left figure shows the age onset of all of the patients in Japan, and this gray bar is the Japanese patients. Their onset was about 28 years old, And these blue bars show the patients with severe mutation, V60603L homozygotes. Their onset was in about 22 years old. In the patients who had mild mutation D207B, their onset was 37 years old. So the age of onset was correlated, differed by genotype. In the patient who has this kind of severe mutation, 50% of the patients of this V603L homozygotes lost ambulance in 10 years, but if they had more mutation in one of the allele, more than 90% could walk more than 20 years. So onset and progression differed by genotype. And and I would like to focus on this small mutation, D20207B. So the frequency of this small mutation was very high, twice as high from the other severe mutation, V603L. But however, this is showing the number of the patients. However, the number of the patients for this mutation, carrying this mutation, was lower than the other mutation. And if we focus on the patient composition, the homozygous patients we could only find 3 homozygous patients. So the frequency of homozygotes were very low. So from the allele frequency in Japan, we could estimate that 240 homozygotes should exist in Japan, but we only could find 3 patients. And we also found a healthy homozygotes. He was 78 years old man, but he had no muscle weakness. So gathering all this information, we estimate that majority of the homozygotes for this mutation does not develop myopathy. And this variant was very far from the catalytic site. And also there were no structure abnormalities of the protein carrying this variant. So this may be reasonable from the structural analysis. And also these 3 homozygotes who only showed mild phenotype had mild reductions in salicylic acid levels. And these characteristics was similar to the founder mutation in India, V727M. So it is previously reported that only 1 homozygote for this mutation exists in 54 Indian patients. And also the patients carrying this mutation had mild phenotypes. So I assume that homozygotes for this mutation may also not develop muscle disease. And also, since some patients who do not have founder mutations, we analyze whether walking prognosis could be predicted regardless to genotypes. And this is showing the probability of ambulation, and this is disease duration. And this red line shows the patients who had their onset in their teens. 50% of these patients will lose ambulance in 9 years, but patients who had their onset in their 20s, 50% of these patients will lose ambulance in 15 years. And in the patients whose onset was there in their 30s, 50% of the patients could walk even 27 years after their disease onset. So we can predict the ambulance loss from the age of the onset. We also evaluated the progression of muscle weakness in skeletal muscle using CT and MRI images from 104 patients. And we assessed the fat replacement using modified Mercury scores. This is a score of 5-point scoring system. And we divided patients into 3 groups based on disease duration, 0 to 5 years, 6 to 15 years, and more than 16 years. And this vertical axis shows names of the muscles, and the horizontal axis shows the modified Mercury score. So this is, if it's low, the muscle is preserved, and if it's number 4, it is the end stage of fat replaced. So if you focus on this 0 to 5 year group, most of the muscle in the trunk are not fat replaced, but muscle in the posterior thigh and lower leg muscles are part replaced. And if we focus on the disease group more than 16 years disease duration, almost all the muscles are part replaced, but quadriceps muscle are well preserved. And if you compare groups categorized by this disease duration, we found that around 5 years of disease duration, the muscles highlighted pink, like posterior thigh muscles or lower leg muscles, or the sum of the pelvic muscle, showed significant fat replacement like this. And in the disease years, about 15 years, these trunk muscles and quadriceps muscles are significantly fat replaced. So this information would be very important to design the clinical trials. So if you include the patients whose onset are about 5 years, you should focus on these pink muscles. But if you want to assess the patients in the late stage, you have to monitor the trunk muscles, like these yellow highlighted muscles. And also we can know which muscle will be affected next, and this may aid to design the rehabilitation program. This is a scheme. So posterior thigh muscle and anterior lower leg muscles would be fat replaced in the middle stage. And in the late stage, even in the late stage, quadriceps muscle is well preserved like this. And I would like to introduce about respiratory function. So generally, it is believed that GNE myopathy patients do not cause respiratory dysfunction, but in Japanese registry, 7 of 220 patients were using ventilator, and low respiratory function was observed, especially after loss of ambulance. And also, respiratory function decline was associated with genotypes. This horizontal axis represents disease duration, and this vertical axis represents the respiratory function. And this red line is the normal respiratory function. So above 80% is normal. So this blue line patients are the patients who have severe mutation in both alleles. They showed decline in respiratory function as disease progressed, but patients who had mild mutation in one side, majority of the patients did not have low respiratory function. And also, we know that diaphragm is the most important muscle of respiration. So I have assessed the diaphragm thickness of left class using CT imaging. So this is the normal diaphragm in the control group. This thickness is normal and their muscle is well preserved. And in GNE myopathy patients who had a low respiratory function, you can see that diaphragm is very thin. And also, this graph shows this is the respiratory function, and this is the diaphragm thickness. And so we can see that thinning of the diaphragm was found to contribute to respiratory dysfunction. So monitoring diaphragm thickness is considered to be very useful for patients who have difficulty doing respiratory function tests. And also we think that diaphragm thinning is one of the reasons of respiratory decline in GNE myopathy patients. And I would like to introduce symptoms commonly associated with GNE myopathy patients, apart from skeletal muscle weakness or respiratory dysfunction. So it was introduced that in the previous presentation, that thrombocytopenia occurs in GNE myopathy patients. And also in our survey, 4.1% of the respondents in our survey were previously or currently diagnosed to have thrombocytopenia, while in Japanese population, the frequency was 0.2%. And it had been reported and hypothesized that hypothyroidism will lead to impairment of cell surface adhesion molecules and increase of platelet-associated IgG will lead to thrombocytopenia. But in our survey, there were no severe bleeding complications of the patient. Still, I think monitoring platelet counts is recommended. And also, there were some patients who had low platelet levels before the onset of the myopathy. So maybe screening the patients who have low platelet levels may aid earlier diagnosis of this disease. Next, I would like to talk about sleep apnea syndrome, as it is reported to be associated with many muscle diseases, such as Duchenne muscular dystrophy or myotonic dystrophy or FSHD. And in our registry data, it was also associated with GNE myopathy. So in our survey, about 16.3% of male respondents and 6.6% of the female patients were previously or currently diagnosed to have sleep apnea syndrome. In contrast, about 3.3% in males and 0.5% of females were diagnosed in general Japanese population. So the frequency of sleep apnea syndrome was higher compared to the general Japanese population. But there were no correlation between patients who had sleep apnea syndrome by grouping in disease duration or respiratory function. So we still do not know the mechanism. But the sleep apnea syndrome is known to be associated with sudden death. It's one of the risk factors. So I think we should check sleep test, especially in the patients who are troubled by snoring or excessive daytime sleepiness. And we also analyze about pregnancy. In our registry, more than 60% of the female patients had experienced pregnancy. And regarding to complications during pregnancy, there were no serious complications, although threatened abortion was relatively frequent in GNE myopathy patients compared to the general Japanese population. This threatened abortion is defined as pregnancy-related bloody vaginal discharge that occurs during the first half of a pregnancy. And threatened abortion raises concerns about preterm delivery. But the frequency of preterm deliveries among patients was not high, and over 90% of deliveries were full-term, and it was as high as the frequency in general Japanese population. So the cases of threatened abortion of the patients in this registry were not severe, and with appropriate management, patients were able to achieve full-term deliveries. So overall, pregnancy in GNE myopathy patients were generally safe. And also we asked about the patients about the impact of disease progression during pregnancy or after delivery. And over 80% of the patients were unaware of the changes in disease progression before, during, or after delivery. Although about 19% of the patients experienced disease acceleration a year after delivery. And in our survey, we identified 6 patients who were first diagnosed to have this myopathy within a year after delivery. So the disease course of GNE myopathy is very variable, and I think it is difficult to examine the correlation with pregnancy about this kind of progression. And maybe the increase in childbearing or housework might lead to a higher awareness of their muscle weakness. Though I think we need a careful observation of muscle weakness, especially within a year after delivery. And also we need further analysis, including sialic acid concentration during pregnancy and after delivery. And at last, I would like to introduce a therapy in Japan. So the big news was that Asenobel was approved in Japan. So in 2009, proof of concept of sialic acid supplementation therapy was established in model mice by Dr. Nishino and Dr. Noguchi in our laboratory. And many clinical trials have been conducted to date. And in phase 2 trial of USA and Israel, there was a maintenance of our upper limb strength. But regrettably, in phase 3 trials, including 7 countries, there are no significant difference, and Autogenics have discontinued the development of this drug. But at the same time, in Japan, we had phase 2, 3 trials, and it showed maintenance of upper limb strength. And also, the same trial was conducted, phase 3 trials conducted, and also it showed maintenance of upper limb strength. And last year, Asenobel was approved. It was 15 years since the proof of concept of the model mice. Last year, it was approved. And this is the result of phase 2, 3 trial of Asenobel in Japan. So in the treated group, the upper extremity composite score was maintained, even though it declined in the placebo group. And I would like to introduce one female patient who responded very well to this drug. So her onset was 27 years old. She noticed difficulty in raising toes. And at the age of 36, she was using ankle foot orthosis and were unable to open the lid of plastic bottle. And she participated in the clinical trial when she was 38 years old. And after participation, 1 month later, she was able to open the lid of new plastic bottle. And 3 months later, she was able to wear trousers. And 6 months later, she was able to walk to the kindergarten about 400 meters. And about 18 months later, she was able to walk to the elementary school about 600 meters. And these improvements in walking ability sustained for 26 months. So this is a report of one case which improvement was observed. And this is a kind of champion data. So not all the patients who took the medication experienced muscle strength improvement. And across the entire trial in Japan, the result was maintenance of muscle strength rather than improvement. So, and we think that it is unlikely that muscle strength would improve in muscle that have been completely replaced by fat. But however, not only this patient, but some patients showed improvement in subjective symptoms and also in objective measurement. So it is possible, I think this is due to the influence of muscles with only maybe mild fat replacement, and maybe this would, some of the muscle symptoms may improve, like in the results of lactose. And also there are some limitations of Asenobel. One is the large tablet size. It's very big, like this €2 coin, and we have to take 12 tablets per day. So it is a bit difficult for the patients with advanced stages. And also it must be taken every 8 hours with meals. If we take it without meals, it would be metabolized more quickly than expected, and it will result in insufficient serum concentration of sialic acid and leading to reduced efficacy. And I really hope that this drug would be available in the patients all over the world. But what is the most regrettable right now is that this Asenobel is now under limited shipping, also in Japan. So it was launched on December 19th last year, but by January 14th, it could no longer be prescribed to new patients. So the company explained that the purification process during the production is very complex, and supply could not keep up with demand. So currently, only 80 Japanese patients who had already started treatment can access the medication, and more than 300 others are still waiting for this to be available for them. And the company is working on revising the manufacturing progress, but it is unclear when this situation can be resolved. And at last, I'd like to introduce the usefulness of antioxidants, which was previously reported by our laboratory. So the pathogenic variants in the GNE gene leads to hypersilation, and this would it would be difficult to remove reactive oxygen species, and there would be increasing oxidative stress, and this will lead to muscle atrophy and weakness. So antioxidant named NAC, this may be available online, was given to the model mice, and the model mice given this drug showed the increase in the distance they can run in treadmill test, and also muscle strength have improved. So antioxidants target a different mechanism from salicylic acid supplementation. So adding antioxidants, maybe we can expect much more further enhance to the effects of muscle maintenance and improvement. Thank you for listening. Thank you.
Thank you so much for that extremely informative talk, Dr. Yoshioka. It was really, really helpful to learn. And also, I think maybe a little bit scary for some of the patients to know all the things that come along with things that we think are not ordinarily told, but your study is finding all of these different other aspects that might impact some people with GNE. So that's really informative, and I think it's really good to have that information. So thank you so much. There are a lot of questions. Saeed Yassin is asking, does GNE affect heart muscles?
No, in our survey, there were no high frequency of heart muscle, and also in many of the reports, cardiac complications are not frequent in the patients.
And you have not done any study on there's this one Indian mutation that's very common, the V727M is the old, but you've not done any study on that. There was one question on that. Neetu's asking, what is the connection between low platelets and GNE? Like that correlation that you're finding, what is the reason for that?
Thank you for the question. So platelet, surface of the platelet is very well sialylated. So hypersialation of the cell surface, the platelet with hypersialation would be attacked, more easily to be attacked. So it's the reason for the low platelets. Adhesion molecules of the cell surface of the platelet is impaired by the hypersialation.
Thank you. So there are a few questions on the Acinobel. I mean, I think you've clarified most of it, but do you have any sense of what the future is looking like for those who are waiting for it outside of Japan?
I'm also asking for the company, but they are too slow to supply. They can, even Japanese patients cannot access to the drug. Majority of the Japanese patients cannot take the drugs. But the company explained that they are now trying to speed up the supply, so speed up the production. So if they could speed up, maybe, and they can supply to Japanese patients, I think they can supply to the other worldwide patients.
So just to clarify to all the patients here, अभी ये दवाई जापान वाली नहीं आ रही है और अभी नहीं अवेलेबल है क्योंकि इसका प्रोडक्शन अभी बहुत ही कम हो रहा है। तो जब ये कोशिश करें कि इसका प्रोडक्शन बढ़ाएंगे, पर अगर प्रोडक्शन नहीं बढ़ेगी तो जब तक नहीं बढ़ेगी तब तक हमें नहीं मिलेगा। और इसके बारे में हम आपको बाद में एक्सप्लेन कर सकते हैं। So there's one question, and also I
Mhm.
think from Michaela, who's also said, great and much needed work on the natural history study. Wakako, many thanks. She's
Thank you
saying,
very much.
she's saying our health technology assessment discussions concluded to set the price of Asinoble. What is the price that you're planning to set?
For Japan, one tablet is about 2,000 yen, so it's about $20 for one tablet. So it's very high. It is covered by under insurance in Japan, but it's a bit high. But yeah, right now it's $20 for one tablet. So we have to take 12 tablets per day. So a bit expensive right now. But maybe they can improve it if they can change the process of the production. If they can produce much more at the same time, maybe they can lower down the price.
Devinder Kaur is asking, is it mostly the case that mutation existing far from catalytic site results in non-symptomatic phenotype?
Not all, thank you for the question. Not all the mutations that are far from the catalytic site shows mild phenotype. So if it's far, it's even though if the mutation is far from the catalytic site, if it is involved in the dimerization or other structural changes, then it will be result in much severe phenotype. But this mutation, it's far from catalytic site and it's far from dimerization surface, and it did not affect any structural changes. And I think maybe with 727M is also have same kind of structural characteristics, but we have not analyzed yet. Thank you.
Thank you so much, Dr. Yoshioka, and thank you so much for being here. In case there are some more questions on the chat, would you be willing to maybe just respond part of this, which is, I would like to invite my mother, Dr. Sudha Bhattacharya, to please. So we are now going to have what we do every time, which is explanation of this entire talk, at this entire conference of the past 2 days in very simple, easy to understand Hindi for those of us who are Hindi speakers, and maybe also those of us who are English speakers but didn't understand. Mark, could you also just, I think there are some people here who are English speakers but did not fully understand the complexity of the talk. Can you put your video on so we can see you? Yeah.
Can you hear me?
Yes. Yeah.
Okay. And I'm visible?
Yes. Yeah.
Okay. So I think everyone is quite tired. We had a very, very productive 2 days of listening to so many talks. So good evening and namaskar. I will try to make it as simple as possible, which is very, very tough. All I can say is that if you have questions, please, you can contact me anytime. So what I will do is that I will speak in Hindi mainly. I know that there are people who are not comfortable with Hindi, and I really apologize to them. What I've done is that slides I've written in English, try to make it simple. So you can read the slides, those who are not able to understand Hindi. And then also later on, you can ask me question, and anytime you can phone me and ask, I can clarify. ठीक है. So, slides are not moving.
Are you trying to move them?
Okay. Can you see it? Can you see the first slide?
Yeah.
ओके। तो पहली टॉक जो मैं बताऊंगी डॉक्टर नलिनी निम्हांस, आप सब पहचानते हैं उनको। निम्हांस का हमारे लिए बहुत ज़्यादा महत्व ये है कि वो पहली हॉस्पिटल है जहाँ पे जीएनई मायोपैथी को डायग्नोस किया गया। तो 2006 में उन लोगों ने ऑलरेडी जीएनई मायोपैथी को उनको पता था, जबकि ये बीमारी इंटरनेशनल भी तभी शुरू हुई थी, उसकी पहचान तभी शुरू हुई थी। तो अभी तक उनके पास बहुत पेशेंट्स हैं और उनका डायग्नोसिस भी बहुत सही होता है। जैसे ही कोई पेशेंट आता है उनके क्लिनिक में तो वो पहचान जाते हैं कि ये किस तरह से चल रहा है, तो ये जीएनई का पेशेंट है। डॉक्टर नलिनी ने हमें ये बताया कि डीएनए डायग्नोसिस ही अब सबसे श्योर तरीका है डायग्नोसिस का। तो बाकी जो टेस्ट हैं, पहले होता था मसल बायोप्सी का टेस्ट, तो अब वो मसल बायोप्सी नहीं लेते हैं आजकल, कम लेते हैं, क्योंकि उसमें पेशेंट को ज़्यादा तकलीफ होती है और डीएनए डायग्नोसिस जब अवेलेबल है तो वो ज़्यादा आसान है। तो ये बहुत इम्पोर्टेंट है कि हमारा डायग्नोज जितना जल्दी हो सके वो हो। अगर आपकी फैमिली में कोई भी है जो आपको लगता है कि शायद उनको जीएनई मायोपैथी हो सकता है, तो आप उन्हें बताइए। तो जैसा आप देख रहे हैं कि बहुत दवाइयाँ अब बन रही हैं और वो जितना जल्दी आप लेंगे डिजीज़ प्रोग्रेशन में उतना उसका लाभ होगा। इसलिए अर्ली डायग्नोसिस बहुत इम्पॉर्टेंट चीज़ है। तो जैसे हम देख रहे हैं हमारे ग्रुप में भी कुछ लोगों का डिजीज़ काफ़ी कम है, कुछ लोगों का ज़्यादा है। तो ये डॉक्टर्स भी देखते हैं यही कि डिजीज़ प्रोग्रेशन जो है वो सब में सेम नहीं होता। कुछ लोगों का बहुत धीरे धीरे प्रोग्रेस होता है और कुछ लोगों का काफ़ी जल्दी होता है। तो इसलिए अगर आपका जल्दी प्रोग्रेशन है तो उसके लिए आप अपने को दोष मत दो, वो आपकी प्रॉब्लम नहीं है। पर ये तो ज़रूर है कि हमारे हाथ में भी बहुत कुछ है और हमें अपने को ठीक करने की भी इच्छा होनी चाहिए। तो एक म्यूटेशन आप लोगों को पता है V77M हमारे देश में सबसे कॉमन पाई जाती है और थोड़ा सा लिंक है कि आपकी क्या म्यूटेशन है और आपकी डिजीज कितनी सीवियर है, इसमें भी थोड़ा लिंक है। पर ये बहुत डायरेक्ट लिंक नहीं है। हम ये नहीं बोल सकते कि आपका ये म्यूटेशन है इसका मतलब आपका डिजीज ऐसा होगा। ऐसा डायरेक्ट इतना स्ट्रॉन्ग लिंक नहीं है। ये डॉक्टर नरीन ने हमको बताया। तो नेक्स्ट टॉक थी डॉक्टर राकेश मिश्रा, जो डायरेक्टर हैं टाटा इंस्टीट्यूट ऑफ जेनेटिक्स एंड सोसाइटी के। तो जब से डॉक्टर राकेश मिश्रा डायरेक्टर बने हैं वो रेयर जेनेटिक डिजीज पे काफ़ी अपने इस सेंटर का उस दिशा में लेके जा रहे हैं उसको। पहले वो नहीं था इतना उनका एम्फेसिस, पर अब वो बहुत ज़्यादा इसमें जोर दे रहे हैं, जो हम लोगों के लिए रियली बहुत अच्छी बात है, क्योंकि वहाँ पे रिसर्च का एटमॉस्फेयर वगैरह सब फैसिलिटी सब बहुत अच्छे हैं। तो डॉक्टर राकेश मिश्रा ने हमें बताया कि वो बहुत चीज़ें कर रहे हैं रेयर डिजीज़ में। एक डेटाबेस बना रहे हैं। वो डेटाबेस में सारे जितने भी उनके पास अवेलेबल डेटा हैं रेयर डिजीज पेशेंट्स के, जितनी स्टडीज हो रही है इंडिया में, वो सब उस डेटाबेस में डालेंगे और फिर वो उसको इंटरैक्टिव बनाएंगे डेटाबेस को, जिससे कि पेशेंट डेटा जो है वो आपका डेटा उसमें होगा और आपको कोई अगर क्वेश्चन पूछना है तो डॉक्टर्स भी वहाँ पे उसमें लिस्टेड होंगे कौन से डॉक्टर अवेलेबल हैं और डॉक्टर्स आपको उत्तर दे सकते हैं और डेटाबेस के थ्रू आपको बहुत नॉलेज गेन हो सकता है। फिर वो डीएनए डायग्नोसिस के ऊपर भी काम कर रहे हैं, क्योंकि जैसा आप बताया लोगों ने कि 7,000 डिफरेंट रेयर डिजीज हैं। तो जीएनई मायोपैथी का डायग्नोसिस तो अब हमको कमर्शियल उससे भी मिल जाता है, पर कई ऐसे हैं जिनका डायग्नोसिस अभी भी बहुत मुश्किल है। तो वो उस पर काम कर रहे हैं अलॉन्ग विद हैदराबाद में जो CDFD लैब है, वो इस चीज़ में बहुत काम कर रही है। थेरेपीज के ऊपर काम कर रहे हैं, तो हम RNA बेस्ड थेरेपी का मैं आपको बताऊंगी, और वो सेल बेस्ड थेरेपीज और स्मॉल मॉलिक्यूल थेरेपी, वो सब के ऊपर भी उनका रिसर्च हो रहा है। अभी उतना बताने लायक नहीं है कुछ, पर धीरे धीरे करके आएंगे रिजल्ट। फिर एक वो रीड्रेस करके एक एनुअल मीटिंग करते हैं। रीड्रेस है सारी रेयर डिजीज के ऊपर। उसमें वो क्लिनिशियंस, साइंटिस्ट, इंडस्ट्री के लोग, गवर्नमेंट पॉलिसी मेकर सबको बुलाते हैं, साल में एक बार होता है। और उसमें क्या है कि जो साइंटिस्ट है वो भी जानता है कि पेशेंट्स के सिम्टम क्या है, वो क्लिनिशियन के साथ इंटरेक्ट करता है। अगर उसके पास ऑलरेडी कोई मॉलिक्यूल है जो ड्रग बन सकता है तो इंडस्ट्री के साथ वो इंटरेक्ट कर सकता है उस मीटिंग में। तो ये मीटिंग अभी तक आई थिंक 3 या 4 साल इसको हुए हैं, बहुत बहुत ही पॉपुलर मीटिंग है। फिर डॉक्टर राकेश मिश्रा अपने एक्सपीरियंस से उन्होंने ये बोला हमें कि अब हम लोग कह सकते हैं कि रेयर डिजीज के एरिया में हमें कुछ उन्नति मिल रही है। पॉलिसी मेकर्स भी अब हमें सुन रहे हैं, और जैसे आईसीएमआर ने अभी सेंटर ऑफ एडवांस रिसर्च शुरू किए हैं जो रेयर डिजीज के ऊपर काम करेंगे। और बहुत अच्छी बात है कि टिग्स को ये स्टेटस मिल गया है, उनको पैसा मिला है टू डू वर्क ऑन रेयर डिजीज थ्रू सेंटर फॉर एडवांस रिसर्च। और जो आईसीएमआर के जो डायरेक्टर जनरल हैं वो खुद ही बहुत इंटरेस्टेड है, और उन्होंने डॉक्टर राकेश मिश्रा को फ़ोन किया पर्सनली और बोला कि अगर आपके पास कोई भी ड्रग बनेगी तो आप बताना, मैं आपको फंडिंग दूंगा फॉर क्लिनिकल ट्रायल। सो दिस इज वेरी वेरी पॉजिटिव फॉर अस। उसके बाद डॉक्टर सीना वेंगलिल की टॉक थी। तो आपको पता है कि वो अभी नेचुरल हिस्ट्री स्टडी में बहुत ज़्यादा हमें हेल्प कर रहे हैं, वही कंडक्ट कर रहे हैं स्टडी। और तो अब उन्होंने समझाया कि होता क्या है नेचुरल हिस्ट्री स्टडी। तो ये स्टडी वो है जिसमें डेटा जनरेट हुआ कि पेशेंट जो जो बीमारी से जूझ रहा है, रोज जूझ रहा है, उसका क्या एक्सपीरियंस है बीमारी के साथ, उसको क्या सिम्टम है, उसका डिजीज में कितना उसको प्रॉब्लम हो रहा है, और कैसे डिजीज बढ़ रही है। ये सब पैरामीटर जो स्टडी करेंगे वो नेचुरल हिस्ट्री स्टडी उसको बोलते हैं, कि नेचुरल स्टेट में डिजीज किस तरह से प्रोग्रेस करती है, वो क्या क्या सिम्टम दिखाती है। क्यों हम ये स्टडी करते हैं, वाई कंडक्ट दिस स्टडी। ये जो डेटा है कि डिजीज कितना सीवियर है, कैसे प्रोग्रेस करता है, ये सब डेटा ड्रग कंपनी को चाहिए जब कोई भी नई ड्रग वो डेवलप करते हैं। इससे वो ये स्टडी उस हमें ये एंड पॉइंट दे सकती है, रिलायबल एंड पॉइंट, कि अगर आप कोई नई ड्रग को टेस्ट कर रहे हैं तो आपको कैसे पता चलेगा कि ये ड्रग काम कर रही है कि नहीं। जैसे हमारी स्टडी में अगर हमें कोई एंड पॉइंट ऐसा मिला कि हमारे ब्लड में कोई चीज़ है जो बढ़ रही है या घट रही है, तो वो एंड पॉइंट हम ड्रग कंपनी को बता सकते हैं कि आप ये यूज़ करो, फिर आपको पता चलेगा ड्रग का इफेक्ट हो रहा है कि नहीं। या फिर एंड पॉइंट ये भी हो सकता है कि कोई मसल का जो है वीकनेस वो बढ़ रहा है, तो अगर हम ड्रग ले रहे हैं तो वो वीकनेस नहीं बढ़ रहा, तो दैट कैन बी ऑल्सो एंड पॉइंट। नेचुरल हिस्ट्री स्टडी जीएनई मायोपैथी में काफ़ी देशों में हुई है, पर इंडिया में ये पहली बार हो रही है। और क्यों ये हमें इंडिया में करना बहुत ज़रूरी है, क्योंकि हमारे जो म्यूटेशन हैं हमारे जीएनई जीन में इंडिया में वो अलग हैं, अब्रॉड जो हैं वो अलग हैं। कुछ कॉमन भी होते हैं पर कुछ बहुत सारे अलग भी हैं, इसलिए हमारे डिजीज़ का हमें खुद पता होना चाहिए। तो ये इम्पोर्टेंट है कि हम अपने पेशेंट के साथ यहाँ पे वो स्टडी को करें। तो नेचुरल हिस्ट्री स्टडी में करते क्या है, तो उन्होंने बताया कि इसमें हम कुछ टेस्ट हैं जो जो हम इस्तेमाल करते हैं मसल की स्ट्रेंथ वगैरह मेजर करने के लिए, और वो स्टैंडर्डाइज टेस्ट हैं जो अब्रॉड भी जो यूज़ किए जाते हैं। वही टेस्ट हम लोग भी इंडिया में कर रहे हैं, क्योंकि अगर हम अपने कोई टेस्ट बनाएंगे तो फिर वो कंपैरिजन हम जो ऑलरेडी स्टडी हो चुके हैं उनसे हम नहीं कर सकेंगे। तो वो क्या क्या टेस्ट हैं, एक तो क्वेश्चनर बेस्ड है। आपको एक फॉर्म दिया जाएगा, उसमें काफ़ी सारे क्वेश्चन होंगे, वो आपको फिल करने हैं। वो क्वेश्चन ये होंगे कि आपको कौन सी एक्टिविटी है जो आप नहीं कर सकते हैं खुद, जिसमें जिसमें आपको किसी की हेल्प चाहिए। और इस तरीके के क्वेश्चन होंगे कि आपकी मूवमेंट और आपकी डेली में जो आप काम कर रहे हैं उसमें आपको किस काम में दिक्कत आ रही है, किसमें एकदम परफेक्ट है, वो सब उस क्वेश्चन में आप बताएंगे। और वो हर 6 महीने में आप उसका उत्तर फिर से देंगे। तो हो सकता है आप 6 महीने पहले कुछ काम कर रहे थे बहुत अच्छे से, अब आप धीरे कर रहे हैं, तो फिर आप बताएंगे कि अब मैं ये धीरे कर रहा हूँ। फिर वो फिजियोथेरेपिस्ट जो है वो आपकी मसल स्ट्रेंथ को मेजर करेंगे, अपर लिम्ब, लोअर लिम्ब, उनके पास मशीन्स हैं, वो उससे मेजर करेंगे ताकि उनको एक डेटा मिलेगा, जिससे कि वो कितनी स्ट्रेंथ है एग्जैक्टली वो उसको एक नंबर देंगे और वो उसको मेजर करेंगे। फिर एमआरआई करेंगे मसल का। एमआरआई से हमें पता चलेगा कि कितना मसल डैमेज हमारा है। जब मसल सेल डैमेज हो जाता है तो उसकी जगह में फैट सेल आ जाता है। वो एमआरआई में दिखता है कि मसल कम हो गई या फैट बढ़ गया। तो इसलिए वो उससे वो देख सकते हैं कि कितना मसल डैमेज हुआ है। फिर वो आपके ब्लड और यूरिन सैंपल्स भी देंगे लेंगे। वो सैंपल्स आपके वो टाटा इंस्टीट्यूट में जाएंगे फर्दर एनालिसिस के लिए। और उस एनालिसिस से हमको, जैसे मैंने बोला कि हमें कुछ एंड पॉइंट चाहिए कि कौन सा मॉलिक्यूल बढ़ या घट रहा है या क्या चेंज आ रहा है हमारे सिस्टम में, वो आपको पता चल सकता है। तो वो ब्लड और यूरिन सैंपल का टेस्टिंग जो है वो टेक्स की लैब में होगा। अभी तक 25 पार्टिसिपेंट्स ऑलरेडी हो चुके हैं और वो 50 कम से कम नंबर हमें करना है, उससे ज़्यादा भी कर सकते हैं। और जो भी स्टडी में जाएंगे उनको 6-6 महीने में जाना होगा और उनका फिर सब पूरा टेस्टिंग होगा। नेक्स्ट टॉक थी डॉक्टर राजेश अय्यर फ्रॉम टाटा इंस्टीट्यूट ऑफ जेनेटिक्स एंड सोसाइटी। तो डॉक्टर राजेश वो mRNA थेरेपी पे काम कर रहे हैं। अब मैं बहुत डिटेल में mRNA का नहीं बता पाऊंगी क्योंकि उतना टाइम नहीं है, पर बहुत ब्रीफली बताऊंगी। आपको पता है कि हमारा जो बीमारी है वो जेनेटिक डिजीज है, मतलब हमारे डीएनए में कुछ डिफेक्ट है। जो जीएनए का डीएनए है उसमें कुछ डिफेक्ट है। तो जो डीएनए का जो डिफेक्ट होता है वो अ, हमारे सेल में प्रोटीन जो होता है, तो प्रोटीन को वो गलत बना देता है। मतलब डीएनए मैसेज देता है आगे मैसेंजर आरएनए को, मैसेंजर आरएनए फिर आगे वो मैसेज को प्रोटीन में पहुंचाता है। तो अगर डीएनए में डिफेक्ट होगा तो मैसेंजर आरएनए में वही डिफेक्ट होगा और प्रोटीन में भी वही डिफेक्ट होगा। और जो प्रोटीन है काम वो कर रही है सेल में, तो अब अगर डिफेक्टिव प्रोटीन होगी तो वो काम नहीं कर पाएगी और बीमारी हो जाएगी। बस ये आपको कॉन्सेप्ट थोड़ा सा ध्यान में रखना है कि डीएनए में डिफेक्ट होगा तो वो मैसेंजर आरएनए में भी होगा और फिर वो प्रोटीन में भी होगा। तो अब ये है कि अगर हम सीधा डीएनए ही दे दें सेल में, जीएनए का डीएनए दे दें जिसमें डिफेक्ट नहीं है, तो फिर वो हमारा करेक्शन हो सकता है। पर हम डीएनए ना दे के मैसेंजर आरएनए भी दे सकते हैं। वो मैसेंजर आरएनए में अगर डिफेक्ट नहीं है तो फिर वो सही प्रोटीन बनाएगा। तो डॉक्टर अय्यर मैसेंजर आरएनए की थेरेपी, एमआरएनए की थेरेपी को डेवलप कर रहे हैं। और वो क्यों एमआरएनए को कर रहे हैं, डीएनए को क्यों नहीं कर रहे, उसका भी थोड़ा मैं बताऊंगी। तो एमआरएनए थेरेपी सेफ है, ये हमको पता चल गया कोविड जब हुआ था। तो सबसे पहली वैक्सीन जो बनी थी अमेरिका में वो एमआरएनए वैक्सीन थी और वो सेफ थी, बहुत लोगों को मिली और सेफ थी। अब एमआरएनए जब हम बनाते हैं वो तो हम लैब में बनाते हैं ट्यूब में, वो हमें बॉडी में इंजेक्ट करना है। बॉडी में इंजेक्ट ऐसे करना है कि वो हमारे मसल तक पहुंचे, तभी हमें लाभ होगा। तो उसके लिए एक व्हीकल चाहिए, वो होता है लिपिड नैनो पार्टिकल व्हीकल। वो आपको इसका चिंता नहीं करना क्या है, पर जैसे वो अपने आप एमआरएनए नहीं जा सकता, उसके लिए हमें कुछ चाहिए जिसके थ्रू वो जाएगा। तो अगर हम नॉर्मल जीएनए का एमआरएनए डाल रहे हैं तो फिर वो नॉर्मल जीएनए की प्रोटीन बनाएगी सेल के अंदर और फिर हमारा मसल सेल जो है वो सरवाइव करेगा। अब एमआरएनए और डीएनए में एक फर्क ये है, एमआरएनए ना उतना स्टेबल मॉलिक्यूल नहीं है, मतलब वो ब्रेक डाउन हो जाता है, जबकि डीएनए बहुत स्टेबल है। तो एमआरएनए स्टेबल नहीं है, इसका मतलब जब आपको इंजेक्शन मिलेगा एमआरएनए का तो नॉर्मल एमआरएनए या नॉर्मल प्रोटीन बना रहा है, पर कुछ दिन या कुछ हफ्तों में वो डिस्ट्रॉय हो जाएगा बॉडी में और आपको फिर से लेना पड़ेगा। इसका मतलब कुछ कुछ दिन में, शायद 2 हफ्ते में, शायद 1 महीने में आपको दोबारा वो इंजेक्शन लेते रहना पड़ेगा। जबकि डीएनए में वो प्रॉब्लम नहीं है, डीएनए स्टेबल है। आप एक बार इंजेक्शन ले लो तो कुछ साल तक आपको नहीं लेना होगा। पर उसके लिए कुछ प्रॉब्लम भी है, वो हम बाद में मैं बताऊंगी। तो इन्होंने क्या किया, अभी हमें mRNA को मसल तक पहुंचाना है। आप अगर उसका इंजेक्शन दे रहे हैं तो वो मसल में पहुंच रहा है कि नहीं, इसके लिए वो जो LNP जो उसका व्हीकल है, जो कार है, सोचो एक कार में वो जा रहा है, वो वहां तक पहुंचेगा। उसके लिए उन्होंने वो स्पेशल LNP बनाया जो मसल में जाएगा, और उन्होंने टेस्ट किया माउस में कि हाँ वो मसल में जा रहा है। अब वो उसी टेस्ट को GNA के साथ रिपीट करेंगे कि अब अगर उसमें हमने GNA का mRNA डाल दिया तो फिर वो मसल में जा रहा है कि नहीं, ये वो अभी टेस्ट करेंगे। तो उनको बहुत बड़ा ग्रांट मिला, जैसे मैंने बताया, वो ICMR से कार ग्रांट। तो अब उस पैसे से उनको काफी सारे डिजीज उन्होंने बोला है वो करेंगे, उसमें GNE मायोपैथी भी एक डिजीज है। तो ये पैसे के साथ अब उनका काम और तेजी से होगा। तो यही होप है कि अब ये काम और तेजी से ये लोग करेंगे। नेक्स्ट टॉक थी डॉक्टर मुनिया गांगुली की लैब से। दिव्या राव ने बोला, तो हम व्हीकल की मैं बात कर रही थी कि हमें व्हीकल चाहिए कुछ भी पहुंचाने के लिए, चाहे mRNA है चाहे DNA है, अपने आप वो नहीं जा सकता। तो हमारे पास कई तरह के व्हीकल होते हैं। एक व्हीकल है वायरल बेस, AAV वायरल वेक्टर, वो अभी डॉक्टर अर्का ने बताया था, उसका अगली स्लाइड में बताऊंगी। पर डॉक्टर मुनिया गांगुली एक और वेक्टर तैयार कर रही हैं अपने लैब में जो वायरल बेस नहीं है। तो नॉन वायरल और वायरल वेक्टर में क्या फर्क है? नॉन वायरल वेक्टर के ये एडवांटेज हैं कि वो बॉडी में रिजेक्ट नहीं होगा। देखिए वायरस क्या होता है, वायरस हमें पता है कि जब वायरस हमें अटैक करता है तो हमें बीमारी होती है। तो बॉडी उस वायरस को रिजेक्ट करने के लिए एंटीबॉडी बनाता है हमारा बॉडी, उससे वो वायरस को हम फाइट करते हैं और वो वायरस खत्म हो जाता है बॉडी से। तो जो AAV है वो भी एक वायरस है, और वो ऑलरेडी हवा में है, हमारी बॉडी में वो जा रहा है, हमें पता नहीं है पर वो जाता है, और हमारी बॉडी उसको रिजेक्ट करती है थ्रू एंटीबॉडी। तो अब अगर मैं AAV वेक्टर से कुछ ले रही हूँ तो वो रिजेक्ट होने का चांस बहुत हाई है। वो चांस नॉन वायरल वेक्टर में नहीं है, क्योंकि नॉन वायरल वेक्टर तो एक केमिकल है, वो तो हमारी बॉडी ने देखा नहीं, तो इसलिए उसका रिजेक्शन का चांस कम है। और दूसरा ये है कि DNA बेस्ड जो है, AAV वेक्टर बेस्ड जो है, उसमें AAV वेक्टर में हम एक ही डोज ले सकते हैं क्योंकि वो एंटीबॉडी बनाता है। सेकेंड डोज लेंगे तो वो बहुत ही फटाफट वो रिजेक्ट हो जाएगा। इसलिए एक ही डोज पहली बार में आपको करेक्ट अमाउंट देना है उसका। जबकि वायरल वेक्टर, नॉन वायरल में वो प्रॉब्लम नहीं है। नॉन वायरल में आपको सिंगल डोज के ऊपर डिपेंड नहीं करना, आप बार बार ले सकते हैं। जैसे आपने एक बार इंजेक्शन लिया और आपको कुछ फायदा हुआ, पर 6 महीने 1 साल बाद फायदा कम होने लगा, तो आप एक और इंजेक्शन ले सकते हैं। जबकि AAV के साथ वो प्रॉब्लम है, एक बार इंजेक्शन लिया उसके बाद दोबारा इंजेक्शन लेना बहुत प्रॉब्लम है। इसलिए नॉन वायरल वेक्टर के कुछ फायदे हैं। तो उसके लिए लोग काफी सारे केमिकल ट्राई कर रहे हैं, डिफरेंट केमिकल्स। तो ये लोग एक तरह का पॉलीमर ट्राई कर रहे हैं जो उन्होंने बताया, PBAE पॉलीमर। वो पॉलीमर उन्होंने लैब में केमिकली बना लिया है, और वो पॉलीमर जो है उसमें DNA के साथ उन्होंने पैकेज कर लिया DNA को, और उन्होंने दिखाया कि अच्छे से वो बन रहा है, DNA उसमें अच्छे से पैकेज हो गया। अब जब हम बॉडी में इसको इंजेक्ट करते हैं तो क्या होता है, बॉडी इसको देखेगी और उसके लिए एक फॉरेन ये ऑब्जेक्ट है, तो उसको वो डिग्रेड भी कर सकता है। तो हमें इस तरीके से बनाना है कि बॉडी में वो तबाह ना हो जाए, वो स्टेबल रहे बॉडी, थोड़ी देर के लिए कुछ टाइम तक स्टेबल रहे, जब तक वो मसल के अंदर नहीं चला जाता। तो हमने जैसे ब्लड में वो गया, ब्लड से मसल में जाएगा, तो उसमें कुछ घंटे अगर लगे, उतने टाइम के लिए वो स्टेबल रहे, वो ऐसे नहीं कि तभी के तभी वो बॉडी में घुल गया और खत्म हो गया। तो वो टेस्ट कर रहे हैं कि कितना स्टेबल है बॉडी के अंदर। अभी तो माउस में करेंगे, बाद में फिर अच्छा रिजल्ट आएगा तो ह्यूमन में। उन्होंने मसल का सेल लाइन में उसको टेस्ट किया है, और मसल सेल लाइन में उन्होंने दिखाया कि प्रोटीन जो GNE की है वो बन रही है, ये उन्होंने दिखाया। अभी वो माउस में अभी स्टडी करेंगे, और माउस में उन्होंने उसको इम्प्रूव करने का की कोशिश करेंगे कि जो डाल रहे हैं वो ज़्यादा देर तक स्टेबल रहे और आगे वो मसल सेल में जाए। क्या होता है जब हम कुछ भी ऐसे इंजेक्ट करते हैं बॉडी में, वो मसल में कम जाता है, लिवर में ज़्यादा जाता है। मतलब बॉडी का मैकेनिज्म है कि सब कुछ लिवर में पहले जाता है, तो हम लिवर में उसको नहीं, लिवर में जो गया तो हमारे लिए वेस्ट हो जाएगा। तो हम चाहते हैं कि लिवर में नहीं जाए और मसल में जाए, उसके लिए वो उसको इम्प्रूव कर। फिर इसके नेक्स्ट में था डॉक्टर अर्क शिब्रा घोष, नारायण नेत्रालय। ये इनको आपने पहले भी सुना हुआ है। तो ये बहुत अच्छा काम कर रहे हैं वायरल बेस्ड वेक्टर के ऊपर। तो वायरस का जैसे मैंने आपको बताया कि इसका कुछ कुछ प्रॉब्लम है, क्योंकि ये ऑलरेडी हमारी बॉडी एक्सपोज्ड है वायरस से, तो इसलिए हमारी बॉडी इसको रिजेक्ट कर सकती है। तो फिर इन्होंने, क्योंकि ये भी मसल डिजीज पे काम कर रहे हैं, जीएनई मायोपैथी, डीएमडी, मसल डिजीज पे कर रहे हैं, तो जो वायरस है दोबारा वही है कि इट शुड गो टू मसल, वो मसल में जाए, ना कि लिवर में या और कहीं पे। उसके लिए वो वायरस को मॉडिफाई कर रहे हैं जिससे कि वो मसल में जाए, और वो डीएमडी मॉडल को टेस्ट कर रहे हैं। अगर वो काम कर गया, मसल में जा रहा है तो डीएमडी में, तो हमारे लिए भी सेम काम करेगा। जीएनई मायोपैथी में उन्होंने कुछ इनिशियल काम किया था, हमने उनको कुछ फंड दिए थे, तो पर हम लोगों के पास इतना पैसा तो कभी होगा नहीं, अब वो उनको गवर्नमेंट फंडिंग से ही वो आगे बढ़ा सकते हैं। तो वो वेट कर रहे हैं कि जब फंड और मिलेंगे तो जीएनई काम को वो और आगे बढ़ाएंगे। उन्होंने जो अभी तक दिखाया है कि जीएनई की प्रोटीन जो उन्होंने बनाई है, वो एबी वेक्टर के थ्रू जब वो माउस में डालते हैं तो वो जीएनई की प्रोटीन बनाता है। जब वो डीएनए को डालते हैं माउस में, वो जीएनए की प्रोटीन बना रहा है और वो मसल में बना रहा है, ये उन्होंने दिखाया। पर आगे और काम उसको करने के लिए उनको फंड चाहिए। उन्होंने बहुत ही वर्ल्ड क्लास फैसिलिटी बनाई है टू प्रोड्यूस एवी वेक्टर, क्योंकि एवी वेक्टर में डीएनए जब पैकेज करेंगे उसका बहुत ही हाई ग्रेड, हाई क्वालिटी का होना चाहिए, नहीं तो वो टॉक्सिक होगा बॉडी के लिए। तो उन्होंने वो बना लिया है, उनको रेगुलेटरी अप्रूवल भी मिल गया है। और अब वो उसको फर्दर ऑप्टिमाइज कर रहे हैं, क्योंकि कॉस्ट को कम करना है। तो ऑप्टिमाइज करने का ये मतलब है कि अगर हम कम वायरस डाल के हमारा काम चल जाए, मतलब वायरस बहुत एफिशिएंट है, बहुत सारी प्रोटीन बना रहा है बॉडी के अंदर, तो फिर हमें कम वायरस में भी हमारा काम चल जाएगा, तो कॉस्ट कम हो जाएगी। इसके ऊपर वो काम कर रहे हैं। उन्होंने बताया कि जो उन्होंने ऑलरेडी डेवलप किया है वो इम्पोर्टेड थेरेपी के हिसाब से 30 to 100 टाइम कम कॉस्ट में होगा जब भी वो उनकी आएगी टेक्नोलॉजी। तो ये बहुत ही होपफुल चीज़ें हैं, पर ये अभी अभी भी ये इनिशियली है, मैं बोलूँगी अभी इसको मार्केट में आने में टाइम लगेगा। इसके बाद जो टॉक थी वो डॉक्टर रवि मंजित आया, वो खुद बोल नहीं पाए थे, उनको बहुत सिवियर लेरिंजाइटिस था, तो उनके स्टूडेंट ने प्रेजेंट किया। वो बैंगलोर में JNCSR में काम कर रहे हैं। तो उन्होंने, वो माइटोकॉन्ड्रियल डिजीज पे काम कर रहे हैं। आप सोचेंगे माइटोकॉन्ड्रियल डिजीज का हमारे लिए क्या है, पर हमारा बहुत लेना देना है उसके साथ, क्योंकि माइटोकॉन्ड्रिया क्या है, वो हमारे सेल में एनर्जी प्रोड्यूस करते हैं। और मसल, आपको पता है कि मसल को बहुत एनर्जी चाहिए काम करने के लिए। तो अगर हमारे जीएनई मायोपैथी में माइटोकॉन्ड्रिया खराब हो गए तो हमारे लिए फिर मसल जो है वो भी खराब हो जाएगा, हमारा मसल। इसलिए हमें ये, हमारे लिए ये इम्पोर्टेंट है कि माइटोकॉन्ड्रिया के बारे में हम समझें। तो डॉक्टर रवि माइटोकॉन्ड्रियल डिजीज पे काम कर रहे हैं, जहाँ पे माइटोकॉन्ड्रिया ही डायरेक्टली अफेक्टेड है। हमारा डायरेक्ट अफेक्ट है मसल के साथ, पर माइटोकॉन्ड्रिया हमारे मसल की वजह से खराब हो रहे हैं और हमारा मसल और भी उसके लिए खराब हो रहा है। पर वो जो कर रहे हैं वो डायरेक्ट माइटोकॉन्ड्रियल बीमारी पे काम कर रहे हैं, जिसमें माइटोकॉन्ड्रिया डैमेज हो जाते हैं और उसके बाद जो सेल है वो मर जाता है। तो हमें पता है कि जीएनई मायोपैथी में भी माइटोकॉन्ड्रिया डैमेज होंगे, क्योंकि माइटोकॉन्ड्रिया के ऊपर प्रेशर आएगा, जब मसल वीक हो रहा है तो माइटोकॉन्ड्रिया के ऊपर प्रेशर आएगा और वो डैमेज होंगे। अब ये डैमेज को हम कैसे रोक सकते हैं, इसके ऊपर वो काम कर रहे हैं। तो क्या होता है कि जब सेल में कुछ डैमेज होता है तो सेल का मैकेनिज्म है उसको वो निकाल देता है सेल से बाहर, मतलब एक तरह से क्लीनिंग अप ऑपरेशन जो होता है, तो घर में गंदगी है उसको निकाल देते हैं। तो वो प्रोसेस को बोलते हैं माइटोफेजी। माइटोफेजी मैकेनिज्म से वो जो डैमेज होते हैं उनको निकाल देते हैं। पर अगर बहुत डैमेज होता जा रहा है तो सेल उसमें फिर वो प्रोसेस उसकी वीक हो जाती है और वो नहीं निकाल पाता। और जब डैमेज माइटोकॉन्ड्रिया बढ़ते जाते हैं तो फिर सेल मर जाते हैं। अब उन्होंने ये ड्रग ट्राई किया यूरोलिथिन ए। यूरोलिथिन ए वो जो ड्रग है वो माइटोफेजी मतलब डैमेज माइटोकॉन्ड्रिया को निकालने की का जो प्रक्रिया है उसको वो हेल्प करता है। और यूरोलिथिन ए का फायदा ये है कि ये ऑलरेडी एक फूड सप्लीमेंट है, ये टॉक्सिक नहीं है, बहुत सस्ता भी है, बाजार में आसानी से मिलता भी है, और ये हमारे बॉडी में बनता है, ये नेचुरल प्रोडक्ट है। तो जब हम ऐसे फ्रूट खाते हैं जैसे अनार या कुछ नट्स वगैरह सीड्स, तो उससे उसमें इसका प्रिकर्सर होता है यूरोलिथिन का, वो फिर बॉडी में जाके यूरोलिथिन बन जाता है। वो हमारे इंटेस्टाइन में बैक्टीरिया होते हैं वो उसको यूरोलिथिन में कन्वर्ट करते हैं। तो ये एक नेचुरल प्रोडक्ट है जो ऑलरेडी हमारी बॉडी को पता है, एक्सपीरियंस है हमारी बॉडी को इसका। पर अगर हम उसको टैबलेट लेंगे तो उसकी क्वांटिटी बढ़ जाएगी हमारी बॉडी में और वो फिर हमको हेल्प कर सकता है। उन्होंने दिखाया कि माइटोकॉन्ड्रिया में यूरोलिथिन ए माइटोकॉन्ड्रियल हेल्थ के लिए उन्होंने मॉडल सिस्टम में दिखाया कि ये हेल्प करता है। तो अगर माइटोकॉन्ड्रियल डिजीज का उनके पास मॉडल है, उसमें यूरोलिथिन ए जब डालते हैं तो माइटोकॉन्ड्रियल डैमेज कम हो जाता है। अब अच्छी बात ये, जैसे यूरोलिथिन ए एक्चुअली कमर्शियली अवेलेबल है, इजीली अवेलेबल है एज अ फूड सप्लीमेंट। तो अब इंडिया में भी एक लैब ने इसको बनाया है और उन्होंने वो इंडियन प्रोडक्ट को टेस्ट किया और वो कह रहे हैं कि ये बहुत अच्छा काम करता है। अब जीएनए में भी ये हम इसको टेस्ट कर सकते हैं जीएनए के मॉडल में, और दिखा सकते हैं कि जीएनए में असर करता है कि नहीं। अगर असर करता है तो फिर ऑलरेडी हमें पता है कि ये ह्यूमन यूज के लिए सेफ है, लोगों ने इसको और चीज़ों के लिए ले रहे हैं लोग, तो हमें क्लिनिकल ट्रायल की परमिशन जल्दी मिल सकती है। पर पहले हमको जीएनई मॉडल में दिखाना पड़ेगा यूरोलिथिन ए लेने से हमारी मसल को कुछ फायदा हो रहा है कि नहीं। उसके बाद जो टॉक थी वो मिखेला ओनाली फ्रॉम इटली। तो मिखेला ओनाली खुद ही जीएनई मायोपैथी की पेशेंट है। उन्होंने हमें ये दिखाया कि पेशेंट्स को अगर साइंटिस्ट के साथ काम करें तो कितना लाभ होता है। तो वो पेशेंट हैं पर उनको पता है कि हमारी इतनी रेयर डिजीज है, साइंटिस्ट को पता भी नहीं है, जानकारी भी नहीं है इसके बारे में, तो इसलिए वो खुद लग गए और उन्होंने साइंटिस्ट के साथ मिल के एक रिसर्च प्रोजेक्ट बनाया और वो यूरोपियन यूनियन की गवर्नमेंट ने उसको फंडिंग दिया, 3 साल के लिए फंडिंग मिला। तो क्यों पेशेंट्स का इन्वॉल्वमेंट जरूरी है, उन्होंने अच्छे पॉइंट बताया कि अगर पेशेंट्स इन्वॉल्व होंगे तो क्यों रिसर्च और अच्छा होगा, क्योंकि पेशेंट को पता है कि हमें क्या चाहिए। जब वो रिसर्च एनवायरनमेंट में आते हैं, साइंटिस्ट से बात करते हैं, वो क्वेश्चन पूछ के साइंटिस्ट को बता सकते हैं कि आप हमें ये दो, हमें ये चाहिए, जिससे कि साइंटिस्ट करेक्ट दिशा में सोचें। और फिर साइंटिस्ट अपने ही लैब में, एक साइंटिस्ट के का बस का नहीं है सब कुछ करना, तो वो पेशेंट मिल के साइंटिस्ट के साथ वो कोलैबोरेटिव रिसर्च के लिए थोड़ा सा ट्राई कर सकते हैं कि 2-4 साइंटिस्ट मिल के कुछ काम करें जिससे कि वो सक्सेसफुल और ज़्यादा हो। और फिर वो रेगुलेटरी बॉडीज के साथ भी, गवर्नमेंट बॉडीज के साथ भी बात कर सकते हैं पेशेंट, जिससे कि अगर ड्रग बन रहा है तो उसका अप्रूवल जल्दी मिले। तो पेशेंट्स की बात लोग ज़्यादा सुनते हैं। अगर कोई भी पेशेंट साइंटिस्ट के पास जाएगा तो वो उसका एक सहानुभूति का वो असर होता है, तो वो सोचेगा ठीक है मेरे को इस पे काम करना चाहिए। उस तरीके से पेशेंट जो है वो एक टीम बना सकते हैं, जो कि मिखेला ने दिखाया कि उसने बनाई टीम यूरोप में और उन्होंने ये प्रोजेक्ट शुरू किया। ये प्रोजेक्ट है एक, हमें पता है कि मैननाक एक ड्रग है जो टेस्ट भी हो रही है, और मैननाक अगर हम लें तो हमारे लिए लाभ है। पर मैननेक का प्रॉब्लम यह है कि जब हम उसको लेते हैं तो बहुत कम वो अंदर जाता है मसल के, और मोस्टली तो वो यूरिन में ही निकल जाता है। तो उसका अपटेक को कैसे इम्प्रूव करें, इसके ऊपर ये प्रोजेक्ट था। तो मैं ज्यादा नहीं बोलूंगी इसके बारे में, यही है कि ये प्रोजेक्ट था कि मैननेक की अपटेक को इम्प्रूव करने के लिए। तो 3 साल हो गए, प्रोजेक्ट अब खत्म हो गया। अभी उनको वो ड्रग तो नहीं मिला जो इम्प्रूव्ड उनको, जिसके लिए शुरू किया था। अ, क्योंकि आपको पता है रिसर्च में कोई भी आप रिसर्च करेंगे तो वो इतना इजीली उसका फाइनल रिजल्ट नहीं आता है, टाइम लगता है। पर तब भी 3 साल के बाद उन्होंने काफी कुछ सीखा उन्होंने, और वो उन्होंने हमको बताया कि क्या क्या सीखा। अ, सबसे बड़ा लाभ तो ये हुआ कि इतने सारे देश यूरोप के जुड़ गए, और उनके साइंटिस्ट एक साथ काम किया, एक टीम बन गया। जो उनमें कुछ साइंटिस्ट है जिन्होंने जीएनई मायोपैथी का कभी नाम भी सुना नहीं था, तो अब वो लोग इसमें काम कर रहे हैं। दूसरा, उन्होंने एक जीब्रा फिश का एक मॉडल बनाया जो कि माउस मॉडल से शायद बेटर है, तो उसको वो यूज कर सकते हैं। और फिर उन्होंने कुछ ये ब्लड टेस्ट में बायोमार्कर भी निकाले हैं। जो बायोमार्कर ये है कि बीमारी कितनी बढ़ रही है, इसको हम उसको मॉनिटर करने के लिए। तो ये अगर अच्छे हैं तो फिर ये बहुत हमारे लाभदायक होंगे। तो अभी उन्होंने और फंडिंग के लिए वो ट्राई कर रहे हैं, जिससे कि अब नेक्स्ट फेज इस प्रोजेक्ट का आगे बढ़ेगा। उसके बाद बहुत इंपॉर्टेंट टॉक थी डॉक्टर पॉल मार्टिन, जो कि अगेन एवी बेस्ड टेक्नोलॉजी में काम कर रहा है। पर उनकी टेक्नोलॉजी जो है वो थोड़ी सी अलग है। वो ड्यूअल वेक्टर टेक्नोलॉजी है, जिसमें 2 जीन को एक साथ वो डालेंगे। एक जीन तो हमारी जीएनई है, क्योंकि हमारे जीएनई जीन का नॉर्मल कॉपी हमें डालना है हमारी बॉडी में। और जो दूसरी जीन है वो है एक जीन जो हमारी जो मसल डैमेज हो गई उसको रिपेयर करने के लिए। जैसे कुछ लोगों ने पूछा था सवाल कि अगर मैं ये जीन थेरेपी लूं तो जो मसल ऑलरेडी खत्म हो गई वो क्या ठीक हो जाएगी? तो वो अगर मैं खाली नॉर्मल जीएनए जीन डालूंगी तो वो जो मसल ऑलरेडी खत्म हो गई वो ठीक नहीं होगी। सिर्फ वो जो हमारी मसल ऑलरेडी कायम है उसको और वो ठीक रखेगा, जिससे कि वो डैमेज ना हो। पर इन्होंने जो किया है, ये 2 जीन डालने से क्या है, एक जीन तो है जीन रिप्लेसमेंट जो जीएनए की नॉर्मल जीन है। दूसरी है मसल बूस्टर जीन, फॉलिस्टैटिन। पॉलीस्टैटिन जीन जो है वो नई मसल को बनाने में हेल्प करता है। तो उन्होंने ये भी डाली है, और उन्होंने दिखाया माउस मॉडल में कि जब वो फॉलिस्टैटिन जीन भी डाल रहे हैं तो माउस में फिर नया मसल, मतलब मसल मास इंप्रूव हो रहा है माउस का। अब वो इसके ऊपर टॉक्सिकोलॉजी स्टडी करेंगे, क्योंकि पॉलीस्टैटिन जीन अगर ज्यादा बन गई बॉडी में, ज्यादा मसल बनने लगा, तो उसका भी कुछ प्रॉब्लम हो सकता है। तो इसके ऊपर वो टॉक्सिकोलॉजी स्टडी करके, और फिर अगेन फंडिंग की प्रॉब्लम है, क्योंकि जीएनए मायोपैथी इतनी रेयर डिजीज है। तो अगर उनको फंडिंग मिल गया तो फिर वो क्लिनिकल ट्रायल के लिए आगे जा सकते हैं। ओके, तो ये तो कल के टॉक्स थे। अब आज हमने डॉक्टर मोनिका पहुजा आईसीएमआर से उनको सुना। जैसा आप देख रहे हैं कि आईसीएमआर जो कि पहले रेयर डिजीज और जीएनए मायोपैथी जो इतनी रेयर डिजीज है उसके बारे में तो वो सुनना ही नहीं चाहते थे। जब हमने इस पे काम करना शुरू किया था तो वो जीएनए मायोपैथी पे फंडिंग मिलना भी मुश्किल था। पर अब जो सीन है वो काफी काफी इम्प्रूव हो गया हमारे लिए। जैसे कि उन्होंने बताया कि आईसीएमआर अब रेयर डिजीज के ऊपर काफी इंटरेस्ट ले रहा है, और फंडिंग उसके लिए देने की उनकी है कि हम इसमें फंड दें और दवाई निकालें। तो एक कंसोर्शियम उन्होंने बनाया है। वो बहुत लंबा चौड़ा नाम है, पर वो कंसोर्शियम में ये है कि साइंटिस्ट, डॉक्टर और इंडस्ट्री के लोग और गवर्नमेंट के लोग जो ड्रग डेवलपमेंट की अप्रूवल देते हैं, वो सबको उन्होंने साथ में जोड़ा है। तो वो उसमें फंड भी दिया उन्होंने कि रिसर्च प्रोजेक्ट जो लोग अगर अच्छे प्रोजेक्ट देंगे तो उसमें वो कंसोर्शियम मिल के डिसाइड करेगा किसको कितना फंडिंग देना है। तो अभी तक उन्होंने कुछ ही रेयर डिजीजेस को इंक्लूड किया बाय नेम, क्योंकि जो भी उनके कारण है वो उतना उन्होंने नहीं डिटेल में दिया। पर जो नाम उन्होंने दिए वो बहुत थोड़े डिजीज हैं, जबकि हमारे पास तो सैकड़ों डिजीज हैं जिसके ऊपर काम होना चाहिए। तो उन्होंने बोला कि पर ऐसा नहीं है कि और डिजीज को वो फंड नहीं देंगे, अगर आप हमें बताओगे तो हम आपको फंड देंगे। वो हमें पता भी है, जैसे टिक्स को तो फंड मिला जीएनई मायोपैथी के लिए। तो अब आईसीएमआर ने काफी फंडिंग अपॉर्चुनिटीज हमें दे दी हैं रेयर डिजीज में। और अब उन्होंने ये मित्रा स्कीम भी शुरू की है, क्योंकि क्या होता है कि रेगुलेटरी अप्रूवल मिलना बहुत ही मुश्किल चीज़ है। उसमें इतना रेड टेप और इतने फाइल इधर उधर डिपार्टमेंट में जाओ, तो लोग परेशान हो जाते हैं। अब उन्होंने मित्रा स्कीम शुरू की है, जिसमें कि अगर आपके पास कुछ है मॉलिक्यूल जो आप सोचते हैं कि ड्रग में कन्वर्ट हो सकता है, या फिर आप कोई नया प्रोजेक्ट शुरू करना चाहते हैं, तो वो आपको हेल्प करेंगे, आपको बताएंगे कि इस तरीके से आप करो तो आपको अप्रूवल जल्दी मिल जाएगा। और फिर उन्होंने सेंटर ऑफ एक्सीलेंस भी खोले हैं 12 सेंटर ऑफ एक्सीलेंस खोले तो हैं 12। बहुत छोटा नंबर है, हमारा इतना बड़ा देश और इतनी पॉपुलेशन, पर जो भी है एक शुरुआत है, अच्छी शुरुआत है। तो इसका हमें भी धीरे धीरे करके लाभ मिलेगा। फिर डॉक्टर सतीश खाडिलकर, बहुत उनका एक्सपीरियंस है जीएनई मायोपैथी भी और बाकी भी मसल डिजीज में, बहुत ही माने हुए और रिस्पेक्टेड डॉक्टर हैं। तो मैनेजमेंट के तौर पे उन्होंने जो हमें बताए पॉइंट, आप लोगों को ऑलरेडी पता है, पर अच्छा है कि और भी ध्यान में रखें। तो एक तो बैलेंस डाइट, उन्होंने बोला कि खाना जो आप खा रहे हैं आपको रियली सोच समझ के खाना है। वजन बढ़ना नहीं चाहिए ज़्यादा, और हमें लगता है कि ज़्यादा प्रोटीन खा लें तो बहुत अच्छा है, पर बैलेंस में खाना है। प्रोटीन तो खाना है ज़रूर, ऐसे नहीं कि कोई प्रोटीन नहीं खा रहे, पर ऐसा भी नहीं कि बहुत सारा प्रोटीन सप्लीमेंट खा रहे हैं, क्योंकि जितना प्रोटीन आप खाएंगे, ज़्यादा एक्सेस खाएंगे, तो किडनी को ज़्यादा काम करना पड़ेगा उसको निकालने के लिए, क्योंकि बॉडी तो बहुत बैलेंस में रहता है। एक्सेस खाओगे तो कोई ना कोई सिस्टम बिगड़ेगा। इसलिए हमें बहुत ज़्यादा प्रोटीन नहीं खाना, और प्रोटीन सप्लीमेंट ना लेके आप डाइट से प्रोटीन लो, वो ज़्यादा अच्छा है। फिर एक्सरसाइज आपको करना है, सारे जॉइंट्स की एक्सरसाइजेस करनी है, और मसल को स्ट्रांग बनाने के लिए रेजिस्टेंस एक्सरसाइज। पर फिर उन्होंने जो बहुत ज़रूरी बताया कि ऐसे नहीं कि अब मैं लग गया तो बहुत ज़्यादा एक्सरसाइज में लग गया, उससे और भी डैमेज हो सकता है। तो ओवर डूइंग नहीं करना, आपको पता होना चाहिए कि मेरी कितनी हिम्मत है, जितनी आपकी स्ट्रेंथ है उन्होंने बोला उसका 80% में आपको यूज करना है, 20% बचा के रखना है। तो उन्होंने बोला जैसे अगर आप 10 मिनट तक चल सकते हैं तो आप 8 मिनट ही चलो। हाँ, तो एक्सरसाइज को करना है रेगुलरली, पर ओवर बहुत ज्यादा नहीं करना। फिर ड्रग्स, तो उन्होंने बोला विटामिन डी और B12 की कमी बहुत कॉमन है, तो वो आप टेस्ट करा सकते हैं, और अगर आपको डेफिशिएंसी है तो आप वो ले लीजिए। अगर आप कोई और मेडिकेशन लेना चाहते हैं, जैसे कि आयुर्वेद, होम्योपैथी वगैरह, तो उसके लिए आप उन्हीं डॉक्टर्स के पास जाओ जिसको मसल डिजीज में कुछ एक्सपीरियंस है। अदरवाइज वो उल्टा सीधा देंगे, उसमें नेगेटिव भी हो सकता है। फिर घर में आपको रिहेबिलिटेशन के लिए, जिससे कि जरूरी है कि आप घर में ऐसे नहीं कि एक ही जगह में बैठे रहे, तो आप पूरे घर में जा सकते हैं, उससे आपको भी अच्छा लगेगा। तो उसके लिए थोड़ा आपको घर को मॉडिफाई शायद करना पड़े, तो वो आपको देखना चाहिए कि आप घर में एक्सेसिबिलिटी है कि नहीं। और सबसे इम्पोर्टेंट मेंटल हेल्थ के पॉजिटिव एटीट्यूड रखना, क्योंकि ये तो बहुत आसान चीज़ है कि हम डिप्रेस हो जाते हैं, तो डिप्रेशन को काबू में लाना और अपने को पॉजिटिव रखना, उसके लिए स्ट्रॉन्ग सोशल सपोर्ट नेटवर्क, मतलब आपके फ्रेंड्स हैं, आपकी फैमिली है, तो आप एक वो होना चाहिए कि जो लोग आपको आपको पसंद आते हैं उनके साथ आप जुड़ें, और आप वैसे काम करें जो जिसमें आपको पॉजिटिव एनर्जी मिलती है। और पेशेंट ग्रुप, जैसे हमारा ग्रुप है, पेशेंट ग्रुप में भी थोड़ा अपने को एक्टिव रखना, जिससे कि और लोगों से भी आप थोड़ा सीख सकें और आपको हिम्मत उनसे भी मिले। उसके बाद ये तो आपको अब ये भी सुने, आपको याद ही होगा उन्होंने क्या बोला। तो 6 साइलेंट लैक्टोज का क्लिनिकल ट्रायल हुआ है, ये ड्रग 6SL जो है ये दूध में वैसे भी नेचुरली पाया जाता है, तो ये हम हमारे लिए नई चीज नहीं है, हमारी बॉडी इसके लिए यूज्ड है और ये हमारे टॉक्सिक नहीं है। तो उन्होंने जो 6SL दिया पेशेंट को, छोटा ट्रायल किया था उन्होंने, उसमें उन्होंने दिखाया कि अपर लिम्ब का एक्चुअली फायदा, स्ट्रेंथ बढ़ रही है, थोड़ा ज्यादा नहीं पर हल्का, और लोअर लिम्ब में और उसका जो प्रोग्रेशन है वो थोड़ा स्लो हो रहा है। तो ये 2 साल का स्टडी था और इसमें प्लेसीबो, जिसको ड्रग नहीं मिल रहा जो पेशेंट को, उसमें इन्होंने 6 महीने ही ऐसा किया प्लेसीबो, उसके बाद सबको उन्होंने ड्रग देना शुरू किया। तो इसलिए उनका जो रिजल्ट आया, मतलब कितनी प्रोग्रेशन स्लो हो रही है, वो उतना इतना स्ट्राइकिंग नहीं था उसमें। ऐसे नहीं कि बहुत ज्यादा उनको दिख रहा है फायदा, क्योंकि हमारी डिजीज तो स्लो प्रोग्रेशन है, 2 साल में इतना पता नहीं चलता। तो अब वो और ज्यादा कर रहे हैं क्लिनिकल ट्रायल, ज्यादा पेशेंट्स को लेके, और 3 साल तक करेंगे, जिससे कि उनको अप्रूवल मिलने में थोड़ा फायदा होगा, अगर उनको कुछ दिखे कि ड्रग का लाभ हो रहा है कि नहीं। उन्होंने बताया कि वो कंपनी न्यूराजीन, जो कोरियन कंपनी है, उसके साथ वो काम कर रहे हैं। वो क्लिनिकल ग्रेड 6 आयरन लैक्टोज बना रहे हैं, जिसको वो क्लिनिकल ट्रायल में यूज करेंगे। बाकी जो कंपनी जहाँ से हम लोग ले रहे हैं, उन्होंने बोला हमें उसके बारे में कुछ मालूम नहीं है, आप लोगे तो अपने उससे लो, हमें नहीं मालूम। पर 6SL क्योंकि लोग वैसे भी खा रहे हैं, तो टॉक्सिक तो नहीं है, इतना तो हमें पता है। इसके आगे उन्होंने कोई रिस्पॉन्सिबिलिटी नहीं ली कि अगर उसमें कुछ और प्रॉब्लम हो रहा है तो ये हमारे ऊपर है। फिर उसके बाद जो लास्ट टॉक है, वकाको योशियोका, ये भी पेशेंट है और ये रिसर्च भी कर रही हैं। बहुत ही रिमार्केबल चीज़ है कि जीएनई मायोपैथी पेशेंट होते हुए इन्होंने अपना रिसर्च लैब में काम भी कर रही हैं। तो इन्होंने जापान के बारे में हमें बताई कि जापान में जितने भी पेशेंट हैं, 60% नंबर ऑलरेडी एक रजिस्ट्री में है। तो रजिस्ट्री में उन पेशेंट्स का सारा डेटा है और हर साल वो डेटा को अपडेट करते हैं। जैसे कि हम नेचुरल हिस्ट्री में कर रहे हैं कि अब 6 6 महीने में हम अपडेट करते रहेंगे, कि अगर आपका कल आप कुछ काम कर पा रहे थे, अभी आज नहीं कर पा रहे, किस तरीके से सिम्टम चेंज हो रहे हैं, वीकनेस बढ़ रही है, वो पूरा इनका ऑलरेडी जापान में कई साल से उनका ये चल रहा है। एवरी ईयर पेशेंट्स का डेटा अपडेट होता है, तो इसलिए उनके पास ये पूरा डेटा अवेलेबल है और कोई भी ड्रग कंपनी अगर ड्रग बनाना चाहे तो वो डेटा यूज कर सकते हैं। फिर उन्होंने म्यूटेशंस का बताया कि जीएनए जीन में जो अलग अलग म्यूटेशंस हैं, उसमें एक जापान में जो कॉमन म्यूटेशन है D207V, वो अगर होमोजाइगस है, मतलब आपके हमारी बॉडी में 2 क्रोमोसोम होते हैं हर उसके, तो अगर दोनों में ही ये यही म्यूटेशन है, तो फिर जो बीमारी है वो बहुत ही माइल्ड होगा, ज्यादा सीवियर नहीं होगा। और हमारे देश में एक बहुत कॉमन म्यूटेशन है V77M, तो उसका भी कुछ ऐसे ही है। अगर दोनों में वही होगा तो फिर माइल्ड बीमारी या तो दिखेगी नहीं और या फिर बहुत माइल्ड होगी। अब उन्होंने इम्पोर्टेंट चीजें ये बताएं कि क्योंकि जापान में बहुत ज्यादा इन लोगों ने स्टडी किया जीएनई मायोपैथी को, तो उन्होंने देखा कि कुछ पेशेंट्स में, जैसे 4% या 6% पेशेंट में रेस्पिरेटरी प्रॉब्लम भी हो सकता है, पर इतना कॉमन नहीं है, इसलिए हमारे देश में वो अभी तक लोगों ने इतना दिखाया नहीं है स्टडी करके। उन्होंने दिखाया कि कुछ स्मॉल परसेंटेज में होता है रेस्पिरेटरी प्रॉब्लम। और थ्रोम्बोसाइटोपिनिया, जो जो एक ब्लीडिंग डिसऑर्डर है, थ्रोम्बोसाइटोपिनिया में प्लेटलेट काउंट कम हो जाता है तो ब्लीडिंग होती है, वो भी होता है जीएनई मायोपैथी में। क्योंकि वो इसलिए होता है क्योंकि उसको सियालिक एसिड की डेफिशिएंसी जो जीएनई मायोपैथी में है, उससे थ्रोम्बोसाइटोपिनिया भी हो सकता है। वो ऑलरेडी नोन चीज है, पर हमारे पेशेंट्स में वो नहीं है। हमें पता है कि प्लेटलेट काउंट हमारे मोरलेस नॉर्मल है, पर कुछ पेशेंट्स में है। तो हमें इसका पता होना चाहिए, क्योंकि जब भी हम ब्लड टेस्ट कराते हैं तो प्लेटलेट काउंट को हम टेस्ट कराएं और मॉनिटर करें कि हमारा प्लेटलेट नीचे तो नहीं आ रहा। और स्लीप एपनिया भी उन्होंने बताया, वो भी एक प्रॉब्लम हो सकती है। तो इसके बारे में हमें थोड़ा दिमाग में रखना चाहिए कि अगर ऐसा कुछ है तो ये जीएनई से जुड़ा हो सकता है। फिर जापान की जो ड्रग है वो ऑलरेडी जापान में अप्रूवल मिल गया। वो कंपनी एस नोबेल जो बना रही है ड्रग को, 2024 में उनको अप्रूवल मिल गया और ड्रग अवेलेबल है जापानीज पेशेंट के लिए। पर जैसे उन्होंने बताया, कंपनी जितनी डिमांड है उतना सप्लाई वो नहीं कर पा रहे हैं। तो अभी जापान में भी पेशेंट वेटिंग लिस्ट में है दवाई को पाने के लिए। तो इंटरनेशनल मार्केट में अभी उनको टाइम लगेगा। जब तक वो स्पीड अप नहीं करेंगे प्रोडक्शन, तब तक हम लोग को नहीं मिल सकेगा। तो आई थिंक यही मेरी लास्ट स्लाइड है। हाँ, ओके। तो ओके, मैंने बहुत ज्यादा बोला है। तो अगर कुछ क्वेश्चन है तो मैं जरूर आंसर कर सकती हूँ।
किसी को कोई क्वेश्चन है? Thank you, Ma, for that very comprehensive summary.
I have to speak very fast. I'm sorry, because otherwise we'll be sitting here the whole evening. So अगर बाद में कुछ आएगा दिमाग में तो पूछ लीजिएगा।
काफी क्लियरिटी दिया आंटी आपने, मतलब इन शॉर्ट्स काफी समराइज कर दिया। एक बस रिक्वेस्ट था, जैसे हम लोग अभी काफी लोगों में, मतलब जैसे खाडिलकर सर ने काफी फोकस किया वेट मैनेजमेंट वगैरह या फिर प्रॉपर इनटेक, तो हम लोग कोई ऐसे कोई कॉमन सेशन ले सकते हैं इसके ऊपर, सो ऑल पीपल विल गेट अलाइन। बिकॉज मतलब मैंने कुछ डॉक्टर से यहाँ पे अप्रोच किए थे पुणे में वगैरह, तो मुझे वो कॉन्फिडेंस दिखा नहीं, मतलब उनको, मतलब एज ए मस्कुलर डिस्ट्रॉफी बैकग्राउंड वगैरह उनको, मतलब जैसे हमें कुछ क्या प्रोटीन देना चाहिए, सो कैन वी टेक दैट वन सेशन इफ पॉसिबल?
Yeah, I think we'll organize more smaller sessions जिसमें हम लोग स्मॉल ग्रुप में ये सब चीजें डिस्कस करेंगे। मे बी डिफरेंट प्लेसेस विल डू इट, अभी सोच ही रहे हैं, एंड विल गेट बैक टू यू।
यस अंकल, यस, थैंक यू।
Dr. Vishnu also said great summary. So,
We move to the last one.
और किसी के कोई questions हैं? Yeah, I think we can conclude.
Yeah, first thing I would really like to thank all our speakers. They came on weekend at odd hours, stayed with us, and being a great support and encouragement for WWGM. And also the speakers pointed out all the development that is happening. They're very, very encouraging compared to last time. There are a lot of hope, a lot of possibilities that are there, and we can see that from both national and international, everybody, whoever we invited, participated into this. We also want to thank all of you, all the patients, scientists, other people who have participated, doctors, your patience and everything. You sat through uh, almost like 17 hours or so of 15, 16 hours of sessions yesterday and today. And I'm sure you're very tired and you're still assimilating it. And I think the videos will go into, after some editing, into the YouTube channel. So thank you very much for being here, and I really look forward to you. And personally, I want to thank Shilpi for doing a great job in conducting this workshop. She sat through the whole thing yesterday, today, and has done a wonderful job. I don't think any of us could have done this this so well. And we want to thank Shilpi, convey our thanks to your all the interns who have worked behind the scene to make it a success. Um, Again, thank all of you, all speakers, all audience participants, people who ask questions. all interns. Thank all of you to do that. And thank you. And let's hope that next time we'll be able to bring you much more positive results. And I must say that many of our speakers were not feeling very well, and they still came and participated in this thing. Somebody lost his voice, somebody was running high temperature. So it's amazing for your support, and really thank you for all this.
Yeah, I'll say that, it's for all of us. So, as a community, we all have to, benefit from it. And I think this time, a lot of patients have joined, so it's very nice. Otherwise, we are not doing it only for us, right? So it's important that all of you join and we benefit from, meeting with doctors, even if virtually. So I hope quite a few questions of the patients were answered this time, and you are getting more and more confident.
Okay, so please keep visiting our website and write to us if you have anything. We can definitely send your questions to the doctors, and they would participate in anything that is coming up. If there is any kind of problem, you write to us. We'll handle it, whatever possible from us. And thanks everyone. Thanks all our doctors, speakers, etc. All right. Good night, and all of you have a happy Holi.
Yeah. Bye bye.
Bye.
Thank you so much to everyone for joining in